hi, 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 hi. Hi, 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 hi. Hi. It's Science Friday. It's Science Friday. And guess what we're doing? We're doing our favorite thing that we did last week, which was going through our microbiology textbook, which is available at openstacks.org. As always open access textbook available anytime at open at here. Don't sue me, OpenStax. Please don't. I'm just trying to spread the knowledge. We are no. We are not cooking meth. We're not cooking meth. We are reading microbiology textbooks. No meth. So I remember what we last week when we went through this textbook. We went through the first chapter and then half of chapter 2, and the first chapter was all about microbiology, like history. We learned all about how the different kinds of cells, the different kinds of animal kingdoms and whatnot. It's kind of interesting, but it was way more like a history lesson. Not much science, but it was interesting history lesson at least. And then chapter 2, it went like, I gotta say, it went kind of downhill. We started getting way into what a microscope is, and oh my gosh, I didn't realize how many different microscopes there are. There's so many different kinds of microscopes. I knew about some of these, but I've never heard about a few of these. And they're so complicated and technical, and there's so much physics involved, and also they went very deep into like how they're able to see things and magnify, and yeah. Okay, I'm not trying to be a science hater. Let's get that straight. Let's roll back. Science is really cool, and that's why I'm doing this to begin with, but there's some topics of science that maybe are not as fun as other topics of science. And I gotta say, the history of microscopes and different kind of microscopes are in there, but it's also important to know. You need to know kind of how microscopes work, like we went over last week in chat, to combat scientific disinformation. People are convinced they know how things work, and it's completely wrong, and so you need to hit them with how the physics of a microscope when you see that disinformation. There's like the people who think a mirror is a sentient being that will look at you instead of not understanding that it's just a light reflecting. So you gotta hit them with microscopes are real. And it was actually interesting. We were talking a lot about scientific disinformation last week, and I saw a tweet talking about. I guess it's not even just one tweet. It was a gaggle, a herd of tweeters talking about how they don't think microorganisms exist because they can't see them. And I just went, what? What are you talking about? You can literally see them. We went. If they just caught my reading last week, they in the 1600s they were just putting lenses together and going, oh, what the frick? There's little critters moving around in my saliva. Anthony van Leeuwenhoek did all this, and in the year 2025, there's some guy using his phone and saying microorganisms don't exist. He's pretending the wee little beast. They're pretending the wee little beasties, the animalcules, aren't a thing. Blows my mind. Anyway, as I always say, don't fight those people. You could try to educate them, but I think when they're in a herd of like misinformation, I don't know if it's like it's a good idea to even engage. I don't think they're gonna be very receptive. So it's usually best to just go, huh, that's strange, and then move it. Keep it chugging. So I think we left off on this giant graph that went over each of the kind of microscopes there are. So, you know, let us start with understanding again what the microscopes we saw were. So we went over the light microscopes, which have magnification up to 1000x, and this is with visible or ultraviolet light to produce an image. So you got your bright field microscope, commonly used in a wide variety of laboratory applications as a standard microscope, produces an image on a bright background. Bacillus sp showing endospores. So yeah, I feel like this bright field is one of the most common ones. This is for sure the thing, like, if you took any kind of biology lab class or high school biology of some kind, you probably use this. Dark field increases contrast without staining by producing a bright image on a darker background, especially useful for viewing live specimens. Hi Ripko, thank you for checking in. Then there's the phase contrast. Uses refraction and interference caused by the structures in the specimen to create high contrast, high resolution images without staining, making it useful for viewing live specimens and structures such as endospores and organelles. And that's Pseudomonas. I can't say I don't think I have used phase contrast. Honestly, like, my microscope usage is way down. I've barely ever used microscopes. I'm not, like, I always think it's kind of funny. There's some classic things you see when you think about a scientist, and this imagery is used often too. Like, it's usually the microscope and some flasks and some test tubes. And I feel I really didn't use many of those in much of my 10 years of science. Learning is good. Even if you don't want to learn, you know, you can listen to the yapping and just tune it all out. And so I think the test tube imagery is probably one of the funniest things. I never use test tubes. I don't really know who's using a test tube in current science day. But every time, even science, this is even a problem. Not a problem. I'm not trying to start trouble. This is even a thing with science VTubers. They'll have some test tubes tied to their thigh, and I'm like, okay, but who's using that? Where's the conical tubes? Where are the conical tubes at? The only time I think I used a test tube was for flow cytometry. That was about it. And you don't cork it. The part where you have it corked too is, I think, really funny, because who in present day is using the cork? Who's putting a cork in their test tube sample where things could the cork cells can be fallen into your sample? We're wrapping that up in we're not corking anything here. No. Oh yeah, there's the white coat, the safety goggles, and they also have usually an ID badge. I'm not attacking any science VTubers. I think, you know, the image works. It's recognizable for a reason. The safety goggles on top of their head, the PP. It's funny because it's you got PPE kind of, but they never have gloves on. Where are my glove VTubers at? Where's the glove VTubers at? Where are your gloves, guys? You can't just be doing all your scientific duties, having goggles on and a lab coat, and you're not wearing your gloves. Come on. I got distracted. What were we at? Differential interference contrast, the DIC. Uses interference patterns to enhance contrast between different features of a specimen to produce high contrast images of living organisms with three-dimensional appearance, with making it especially useful in distinguishing structures within live, unstained specimens. Images viewed reveal detailed structures within cells. I don't think I've ever used this microscope either, but this image is very recognizable. When it has like this little, it looks kind of like it's popping out of the surface. That's I usually know it's that kind of microscope. I didn't even actually know its name. You do go through a lot of gloves a day. Oh my gosh, one thing I think about with science especially is it's such a wasteful, it's a really wasteful field. There would be a time when I would like look at my pile of what I threw into the garbage with just one day of experiments, and it would be like a bunch of gloves, because I had to go in and out of the rooms, and so you have to, I have to toss my gloves, or I'm touching different things, and I don't want to like cross-contaminate. And a giant mountain pile of pipette tips, plastic pipette tips, so many different like containers that are like one use only, because molecular biology is very, very sensitive to any kind of contamination. And I would stare at the mountainous pile of plastic garbage that I produced in one day, and I went, wow, this is the worst career possible. We should not all be scientists. Okay, but science is really good, but it sucks how big waste it is. Big waste. Big waste. I know there's like a lot of labs that have tried to minimize how wasteful it is, especially with regards to pipette tips. I know some labs try to be mindful, like, do we really need to only use them once? Maybe we can try to autoclave them, especially if you're not doing molecular biology. It's just I think molecular biology is a huge waster because of just how sensitive certain things are, and also how bad cross-contamination or contamination of any kind can be. And also like the tiny Eppendorf tubes. My gosh, you just go through so much. Nowadays I just feel bad when I buy something and it's it comes in like a single-use tear plastic. But when I was a scientist, I'm like, yeah, I need to use a whole box of pipette tips right now. Fluorescence microscopy. Uses fluorescent stains to produce an image. Can be used to identify pathogens, to find particular species, to distinguish living from dead cells, or to find locations of particular molecules within a cell. Also used for immunofluorescence. Yeah. I mean, fluorescence is fluorescence. That is true. I know a lot about fluorescence microscopy. I didn't really get to do this, though. Confocal microscopy uses a laser to scan multiple Z-planes successfully, producing numerous two-dimensional high-resolution images at various depths that can be constructed into a three-dimensional image by a computer, making this useful for examining thick specimens such as biofilms. Example, E. coli stained with an acridine orange dye to show the nucleoid regions of the cells. Oh, I see. You can see the funky little guys right there. So all that white is the nucleoid region. Kind of neat. I think it was really... I never did confocal microscopy, but it was pretty hard to get an appointment. Or, not an appointment. A time slot to get into the confocal... To get, like, access to the confocal microscope. At least in my university, I think it was just... It's just a really popular, It's really popular methodology, I guess. I am not familiar with it, so I can't explain to you what's going on. 2-photon uses a scanning technique, fluorochromes, and long wavelength light such as infrared to penetrate deep into thick specimens such as biofilms. Mouse intestine cells stained with fluorescent dye. Oh, that looks so cool. These are like the things I feel like you see with like a nature journal. Like this is like This is like the cover of one of the big science journals. Like every other month, that's what I feel like they're showing me, and I'm like, interesting, very interesting. That is science. Yes, that is science. There's green, red, and blue. And so those were all the light microscopes, and my experiences with them, my personal one-on-one experiences with them, ends with the bright field microscope. Electron microscope. Now this is even much more far removed from me. I don't even know where I would have, like, when I think about my lab, I don't even know where I would have gotten access to an electron microscope. I'll be real. 20 to 100x, so super magnified. Look how, like, you can tell just how different this is than the light microscopes. Like, compare what you're seeing with the bright field one with what's going on over here in the scanning, the SEM one. Like, you're starting to look at something that almost looks like it's like been 3D. It's like a 3D image, like someone generated. But no, it's just a microscope picture. It's kind of cool. What type of microscope are you even using to be able to stream? You wouldn't understand what microscope I'm using. It's far beyond what anything in this textbook is going to tell you. It's too advanced. I can't go over it. It would take just too secret. Just know there's a lot of like 2D images being shot my way to create this. Well, it's not a 3D image you're looking at. yeah, you wouldn't be able to comprehend what's going on. So there's a transmission electron microscope. It uses electron beams to pass through a specimen to visualize small images, useful to observe small, thin specimens, such as tissue sections and subcellular structures. Ebola. It's all gross. It's kind of gross. So advanced, even I don't know? Yes. The people, it's far beyond me. I'm just the microbe here. I don't make the rules. They wanted to use the microscope on me and put me on show and display. Here I am. I don't know what's going on above me. Scanning. Uses electron beams to visualize surfaces, useful to observe three-dimensional surface details of specimens. Campylobacter jejuni. Oh, campylobacters. Those are the nasty guys. That's the you're gonna be sick and puking if you get this in your tummy. I think this is also like, I remember when I would go to the mouse rooms, there would be so many signs saying like, this place has campylobacter, be careful. If you're pregnant, don't enter this room. There's campylobacter in here. And then these microscopes are the super advanced ones that I actually had no idea even existed until last week we went over these. I only knew about light and electron, and then all of a sudden this textbook threw a curveball and said there's something called scanning probe microscopes, and I said, what? What? We've got these? And this is magnification of 100 to 100 million times or more. And it uses very short probes that are passed over the surface of the specimen and interact with it directly. And I thought it was kind of crazy how advanced this was. I like the fact that it interacts directly with the surface. I was like, what? You can do that? Because when you think about it, all these other microscopes, they're more just like, they're taking a bunch of pictures. They're not really interacting with it in a way. It's just a bunch of pictures. Even this 3D one, it's not like it's interacting. It's just they took a picture from a bunch of different angles, and they're able to recapitulate this into a 3D form. But the scanning tunneling scanning probe microscope. It uses a probe passed horizontally at a constant distance just above the specimen while the intensity of the current is measured. Can map the surface of structures at the atomic level. Atomic! Can you see? It's the atomic level. It's not even like molecular. It's looking at it, the atoms of it. Hello, Sergeant 10. Yeah, we're going through a microbiology textbook. Right now, we're learning all about microscopes. This works best on conducting materials, but can be used to examine organic materials such as DNA if fixed on a surface. So this is just gold. I don't actually know. I remember we were trying to figure out, like, what does this look like for a living organism, and we couldn't figure out any examples. It just looks like it's more common for inorganic things, like gold. Yeah, science. And this is atomic force, and I also thought this looked cool. It kind of looks like... I was like, what's even going on here? Kind of looks like hash browns. Like, they like opened up a hash brown really, really closely. But so this one can be used in several ways, including using a laser focused on a cantilever to measure the bending of the tip, or a probe passed above the specimen while the height needed to maintain a constant current is measured. Useful to observe specimens at the atomic level and can be more easily used with non-conducting samples. AFM height image of carboxymethylated nanocellulose adsorbed on a silica surface. I don't even know what they're... I don't know what they're looking at. But, you know, yeah, sciencey hash brown. I can't tell you what that means, like what they wanted to examine, but that's really, 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 really close up if they're able to see it like that. Let me zoom out again. Okay, so now that was a review from what we did last week, which we finally finished going over all the microscopes. I think this microscope chapter is still not yet over. We still have a lot to conquer. And now we're going through how we stain our samples to use the microscope. So, staining microscopic specimens. By the end of this section, we'll be able to differentiate between simple and differential stains, describe the unique features of commonly used stains, explain the procedures, and name clinical application for gram, endospore, acid-fast, negative capsule, and flagella staining. Hello! This is hold on. This is the OpenStax ASM Press's microbiology textbook. Available for free, open source at openstax.org. Written by it's Rice University's open source microbiology textbook. Free to use, by attribution, free to stream, free for media usage. It's really interesting. We went through chapter 1 so far, and we're halfway through chapter 2. Yeah, that I was surprised to find that. So now we get to have a fun session where I just read this to you while I learn and you learn, and we all sit and learn microbiology together. I see. We're almost at the end. We're almost at the end of this microscope chapter. This microscope chapter it's kind of a drag. I'm not gonna lie, but we're almost there. We're almost free. No. You don't understand. You are learning. You might not realize it, but deep in the recesses of your brain, as you listen, you'll suddenly remember if someone talks about a specific type of microscope, your brain will recall the scanning tunneling microscope. You watch. Just watch. One day the scanning tunneling microscope will come up in conversation. In their natural state, most of the cells and microorganisms that we observe under the microscope lack color and contrast. They are not Yes, microorganisms are not as colorful as me. You know, I'm very colorful. I'm pink and green and blue and all. Yeah, but most things they don't look anything like me. Kind of sucks for them. This makes it difficult, if not impossible, to detect important cellular structures and their distinguishing characteristics without artificially treating specimens. We have already alluded to certain techniques involving stains and fluorescent dyes, and in this section we will discuss specific techniques for sample preparation in greater detail. Indeed, numerous methods have been developed to identify specific microbes, cellular structures, DNA sequences, or indicators of infection in tissue samples under the microscope. Here we will focus on the most clinically relevant techniques. I'm gonna guess they're just gonna talk about things like, you know, fixation and maybe do a bit of like, I guess fluorescence talk, but fixation is probably the biggest one. Yeah, preparing specimens for light microscopy. In clinical settings, light microscopes are the most commonly used microscopes. Yeah, that's what I was saying. That's exactly what I was saying. I'm too dumb to remember gram-negative versus gram-positive. Oh, don't you worry. I'm sure we're gonna go over that in this chapter, probably. I also mix that up a lot, too, though. Honestly, I'm just kind of bad at memorizing a lot of, things. I'm a fake microbe. I don't know my own history. You ask me something, and I probably will be like, And it's not that I'm sure I know it deep in my brain. I just have a hard time pulling up information like that. My memory access is not there. In clinical settings, yeah, the commonly used microscope. There are two types of preparation to view specimens with a light microscope: wet mounts and fixed specimens. The simplest type of preparation is the wet mount, in which the specimen is placed on the slide in a drop of liquid. I did this. I know how to do this. I've done this. I think this is the one technique I feel like most people tend to know how to do, because it's pretty easy. Some specimens, such as a drop of urine, are already in a liquid form and can be deposited onto the slide using a dropper. Oh yeah, if it's already liquid, you just literally put it right on the slide. And then you put the top on. Solid specimens, such as a skin scraping, can be placed on the slide before adding a drop of liquid to prepare the wet mount. Sometimes the liquid used is simply water, but often stains are added to enhance contrast. Once the liquid has been added to the slide, a cover slip is placed on top and the specimen is ready for examination under the microscope. Easy, easy. I think we can all do that. I think if I gave everyone right now a slide, you guys could do that. I believe in you guys. The second method of preparing specimens for light microscopy is fixation. The fixing of a sample refers to the process of attaching cells to a slide. Fixation is often achieved either by heating, heat fixing, or chemically treating the specimen. In addition to attaching the specimen to the slide, fixation also kills microorganisms in the specimen. No! They're killing me. They're killing me to look at me. Stopping their movement and metabolism while preserving the integrity of their cellular components for observation. They're killing me and looking at me and freezing me. To heat fix a sample, a thin layer of the specimen is spread on the slide, called a smear. That is so funny to see bolded. I don't like. yeah, a smear. That is a technical term for it, yes. The slide is then briefly heated over a heat source. Chemical fixatives are often preferable to heat for tissue specimens. Yeah, I was gonna say I don't think I've ever heat fixed anything before. Chemical agents such as acetic acid, ethanol, methanol, formaldehyde. Go formaldehyde. That's the thing I always work on. Woo, formaldehyde. Or glutaraldehyde can denature proteins, stop biochemical reactions, and stabilize cell structures in tissue samples. None of these things are very good for you, to be sniffing. These things are pretty bad, pretty nasty chemicals. I mean, you just gotta think about it. they are killing the cells, and so you have to be careful. You got like, I remember there would be so many warning signs on our formaldehyde bottle, and I remember my undergrad research advisor. She would always be very like, when you work with formaldehyde, make sure you don't breathe it in too much, and when you work with it, take a step back and like breathe in some normal air. And I was like, okay, yeah, but I kind of get it. She was just being this. She's just trying to be very safe for us, and she cared about us. I appreciate it now thinking back on it. So specimen can be heat fixed by using a slide warmer like this. There's a slide warmer? Never in my life have I seen a slide warmer. Another method for heat fixing a specimen is to hold a slide with a schmear over a micro incinerator. I've never done this one either. This tissue sample is being fixed in a solution of formalin, also known as formaldehyde. Chemical fixation kills microorganisms in the specimen, stopping degradation of the tissues and preserving their structure so that they can be examined later under the microscope. They just took the University of Bristol's YouTube video for that. That's kind of funny. Yeah, this formaldehyde fixation is typically what I did. These two, I don't think I've ever been in a lab or heard of a lab that did this. I don't know how popular that is. Is it? Yeah. I'm just wondering, is it like long-term? Is it possible to have it preserved for that long with heat fixture? Because I feel like the nice thing about the formaldehyde, you would have like a lot of labs, they have like a case of their slides, and they can basically always refer to them because they're fixed. I don't know. I am so unfamiliar with heat fixation. In addition to fixation, staining is almost always applied to color certain features of a specimen before examining it under a light microscope. Stains, or dyes, contain salts made up of a positive ion and a negative ion. Depending on the type of dye, the positive or the negative ion may be the chromophore, the color ion. The other uncolored ion is called the counter ion. If the chromophore is the positively charged ion, the stain is classified as a basic dye. If the negative ion is on the chromophore, the stain is classified as an acidic dye. Okay, I actually don't. We're venturing into like specifics of techniques that I'm familiar with, but I don't know the actual science behind the techniques because no one really teaches you the science behind why you're doing certain things. They'll tell you to stain because they'll tell you to stain and they'll tell you oh, it's because you need to visualize what's going on. But they don't want to tell you why the stain works. Oh, hi, Ro! Thank you for stopping by. So smart. No. Not smart. Not so smart. We're just reading a textbook, and I'm learning while you guys are learning at the same time. So I had no idea there was even it makes sense there's chemistry behind why the staining is how it occurs. Dyes are selected for staining based on the chemical properties of the dye and the specimen being observed, which determine how the dye will interact with the specimen. In most cases, it is preferable to use a positive stain, a dye that will be absorbed by the cells or organisms being observed, adding color to objects of interest to make them stand out against the background. My music ended. Hold on. Where's my Animal Crossing playlist? Animal Crossing with rain noises? 10 hours of Animal Crossing music with rain? Okay. Now we're in maximum cozy. We're in maximum comfy zone. Okay, so we talked about positive stain. However, there are scenarios in which it is advantageous to use a negative stain. Oh, thank you for the follow, Ro. Yeah, I do think teaching, reading out loud is a big one. I used to do this to help study. Oh, no worries. No, no, no. Yeah, I don't have any expectations for anyone to feel like they need to follow or not, but I appreciate it. Thank you. And I think like learning like this, especially it's not like I have any classes anymore or any expectations to know microbiology, since especially I work in cancer biology now. Having no pressure to learn something makes it way easier to learn. But also I know that's not the case for some people. Some people, they really need that pressure. They need, or else they don't care to learn. And that is also understandable. Negative staining produces an outline or silhouette of the organisms against a colorful background. Okay, so these Bacillus anthracis cells have absorbed crystal violet, a basic positive stain. So this is the positive stain. You can see it turned the special color, and you can visualize it more clearly. This specimen of okay. Pronouncing my fellow microbe names are really hard. I'm sorry, microbes. Spinilorchis, a microscopic marine organism, has been stained with rose bengal, a positive acidic stain. She's so pretty. Look at this beautiful pink. She's just like me for real. These B. megatherium appear to be white because they have not absorbed the negative red stain applied to the site. Okay, yeah, so negative stain. It stained the background. Now it's just white. I feel like in most scenarios, I've always used a positive stain. I don't recall many times when we did, like, background staining. But you can see, like, imagine if there's no staining at all. There'd be, like, no way to visualize what's going on. They're just it's not like they're white. That's not the case. It's more just like they just have no color. I know, aren't they pretty? They're so pretty. They even have funny shapes. I think the bacteria shapes, they're just so fascinating. Like, look at this guy. The bee one with the hot pink. It's just so, it's got like such an interesting shape. What's going on there? I don't know. Because cells typically have a negatively charged cell wall, the positive chromophores in basic dyes tend to stick to the cell walls, making them positive stains. Thus, commonly used basic dyes such as basic fuchsia, crystal violet, malachite green, methylene blue, and safranin. Some very interesting names. Typically serve as positive stains. On the other hand, the negatively charged chromophores in acidic dyes are repelled by negatively charged cell walls, making them negative stains. Commonly used acidic dyes include acid fuchsia, eosin, and rose bengal. It would be fun to do more like personal lore, talking about how you guys are able to perceive me based on what dye I'm using. Maybe I will like pocket that idea. Some staining techniques involve the application of only one dye to the sample. Others require more than one dye. In simple staining, a single dye is used to emphasize particular structures in the specimen. A simple stain will generally make all of the organisms in a sample appear to be the same color, even if the same sample contains more than one type of organism. In contrast, differential staining distinguishes organisms based on their interactions with multiple stains. In other words, two organisms in a differentially stained sample may appear to be different colors. Differential staining techniques commonly used in clinical settings include gram staining, acid-fast staining, endospore staining, flagella staining, and capsule staining. Figure 2.41 provides more details on these differential staining techniques. I don't recall what kind of differential staining I did, but these all sound really familiar. I mean, I'm pretty sure I did gram staining and acid-fast staining. Those ones I'm pretty positive. I am very positive, in fact, that I did use those techniques. Not familiar with endospore staining or flagella staining, or capsule staining for that matter. But definitely remember gram stains and acid-fast. Yes, here it is. Time to understand what's gram positive versus what's gram negative. This is the thing I always mix up on. Gram staining. Hold on, I have to take a sip of water. Okay. The gram stain procedure is a differential staining procedure that involves multiple steps. It was developed by Danish microbiologist Hans Christian Gram. It's him. He's Gram. In 1884, as an effective method to distinguish between bacteria with different types of cell walls. And even today, it remains one of the most frequently used staining techniques. The steps of the Gram stain procedure are listed below and illustrated in figure 2.33. I can't believe Gram is Gram. I actually didn't. I didn't know that. What do you mean Gram staining is just named after some guy? How is that always the case? I just end up I should just know this, that if it doesn't seem to make sense where the name came from, it was probably named after some guy. Yeah, he's a celebrity. Oh my gosh, the Danish microbiologist Hans Christian Gram. Oh my god. Oh my gosh. And that was at least it was a European biologist with an easier name. I think the names we were coming across in chapter 1 they were much more difficult for me to pronounce, like Anthony van Leeuwenhoek. First, crystal violet, a primary stain, is applied to a heat-fixed smear, giving all the cells a purple color. Oh, maybe I have done heat-fixed staining if we've done Gram staining. Question mark? Next, Gram's iodine, a mordant, is added. A mordant is a substance used to set or stabilize stains or dyes. In this case, Gram's iodine, it acts like a trapping agent that complexes with the crystal violet, making the crystal violet iodine complex clump and stay contained in thick layers of peptidoglycan in the cell walls. Next, a decolorizing agent is added, usually ethanol or an acetone ethanol solution. Cells that have thick peptidoglycan layers in their cell walls are much less affected by the decolorizing agent. They generally retain the crystal violet dye and remain purple. However, the decolorizing agent more easily washes the dye out of the cells with thinner peptidoglycan layers, making them again colorless. I'm just trying to think, have I done this? I think I did this. Did I? I feel like I've been so far removed from science now. Not far removed from science, but I've been so far removed from experimental work now that I don't even know what I know. Did I do anything? Have I ever done a science before in my life? Finally, a secondary counterstain, usually safranin, is added. This stains the decolorized cells pink and is less noticeable in the cells that still contain the crystal violet dye. Okay, so yeah, we described all the steps. Okay. I knew that gram negative turns pink. I remember that because that's why I said I'm a gram negative. I'm a gram negative bacteria because I'm pink. Oh, I did base science off of my character. I should remember to associate that more. The purple crystal violet stained cells are referred to as gram-positive cells, while the red safranin dyed cells are gram-negative. However, there are several important considerations interpreting the results of gram stain. First. Older bacterial cells may have damage to their cell walls that cause them to appear gram-negative, even if the species is gram-positive. Old always cause problem. Old always cause problem in science. Thus, it is best to use fresh bacterial cultures for gram staining. I am fresh. Fresh, young, fresh bacterial culture. Yes. Second, errors such as leaving the decolorizer on for too long can affect the results. For example, if the decolorizer is left on a slide of gram-positive bacteria for a bit too long, most cells will appear gram-positive while a few will appear gram-negative. This suggests damage to the individual cells or that the decolorizer was left on for too long. The cells should still be classified as gram-positive if they are all of the same species rather than a mixed culture. You know, like, going through these, like, reasons why the staining might not work or doesn't appear as it should, despite the fact that it should appear a certain way, makes me go, yeah, science is so rough. It's just like, it's so, like, there's so many things of like, yeah, it could be because of this. Have you tried this? Oh, or maybe have you considered this? Have you considered this aspect as well? Perhaps your bacteria is slightly old? Did you wait a little too long, maybe? Yeah. Experiments. Experiments are it's just so tiring trying to figure out why something doesn't work. Like, please just work. I know you are gram negative, gram positive. Just do something. Confirm yourself. Confirm. Besides their differing interactions with dyes and decolorizing agents, the chemical differences between gram-positive and gram-negative cells have other implications with clinical relevance. For example, gram staining can help clinicians classify bacterial pathogens in a sample into categories associated with specific properties. Gram-negative bacteria tend to be more resistant to certain antibiotics than gram-positive bacteria. We will discuss this and other applications of gram staining in more detail in later chapters. Ooh, and that's the classic, like the staff. See? So you can see, like, the little purple dudes in this figure. I'm gonna zoom in a bit. So, there's two organisms in this figure. So you have the purple guys. These are the crystal violet ones. That's the staph aureus. And then you have, in the background, it's hard to see because they are decolorized, it's E. coli. And so that's how you handle when there's two. And then you can differentiate the two different organisms in your sample. Very cool. Staining is so fun, so interesting, so interesting. Clinical focus. Oh, this, I forgot. Yeah, there's the staining. It's all I finally remember everything about gram negative and gram positive. And will I retain this information next week? We will see. Clinical focus. So these chapters, they have like a little clinical section, like a little clinical application. No, not a quiz. I need to quiz you guys. I'm the lecturer. You can't quiz the lecturer. I know all this theoretically. Hypothetically speaking, I know everything there is to know about microbiology. I own the textbook. I don't own it, but I have it downloaded. So the clinical focus parts of these chapters, they give you like a little hypothetical situation. And for this chapter, there was a camp counselor, Cindy, who was running in the camp and she scraped her leg, and then it got pus filled and looked really nasty, and she went to the camp nurse, and the camp nurse said, "Ooh, we're gonna send that to the medical lab." And we were all shocked that a camp nurse would send we were all shocked that a camp nurse would go send a sample to a medical lab instead of just saying, "Yeah, here's a band-aid." And so they sent her infected knee scraping to a medical lab, and let's see what they find out. Viewing Cindy's specimen under the dark field microscope has provided the technician with some important clues about the identity of the microbe causing her infection. However, more information is needed to make a conclusive diagnosis. The technician decides to make a gram stain of the specimen. This technique is commonly used as an early step in identifying pathogenic bacteria. After completing the gram stain procedure, the technician views the slide under the bright field microscope and sees purple grape-like clusters of spherical cells. Are these bacteria gram positive or gram negative? Well, if we remember, I'm pink. And you know what pink is? Pink equal gram negative. Purple, not gram negative. They're gram positive. What could she have? We won't know until we go to clinical focus part 4. We're on the edge of our seats. Will Cindy live? Will she still keep her? Will she have her leg by part 4? We'll see. We have to continue. Acid fast stains. Acid fast staining is another commonly used differential staining technique that can be used. An important diagnostic tool. An acid fast stain is able to differentiate 2 types of gram positive cells. Okay, now we're not looking at what's gram negative or gram positive. We're looking at what kind of gram positive cells are there. Those that have waxy mycolic mycolic acids in their cell walls, and those that do not. 2 different methods for acid fast staining are oh, I do remember this, because I remember I didn't know how to pronounce this technique. I'm sorry, I'm trying to focus all my brain energy into doing my best attempt at pronouncing this, because I know it's been said to me. Ziehl-Neelsen. I always called it the ZH technique. And the Quignon technique. Never heard of that guy. Sorry to Quignon. Both use carbonyl fusions as the primary stain. The waxy acid fast cells retain the carbonyl fusion even after a decolorizing agent, an acid alcohol solution, is applied. A secondary counterstain, methylene blue. I remember methylene blue. I did do this. It was messy. Is then applied, which renders non-acid fast cells blue. The fundamental difference between the 2 carbyl fusion-based methods is whether heat is used during the primary staining process. The Ziehl-Neelsen method uses heat to infuse the carbyl fusion into the acid fast cells, whereas the Quignon method does not use heat. Oh, so I lied. I just completely lied. I have heat. I have used heat to fix cells of some kind. I'm just lying. Both techniques are important diagnostic tools because a number of specific diseases are caused by acid-fast bacteria. If acid-fast bacteria are present in a tissue sample, their red or pink color can be seen clearly against the blue background of the surrounding tissue cells. I wonder if I'm an acid-fast bacteria. Maybe we'll have to test that. But I think I would also die if you tested that. So let's not. Using microscopy to diagnose tuberculosis. What a jump. We went from staining to now we're diagnosing tuberculosis. All right, let's get into the application of things. Okay, we learned how to stain the bacteria. Now we're gonna go detect tuberculosis. How are we doing a textbook? What are we doing textbook? Mycobacterium tuberculosis. Oh, if you watched my stream where I went over my academic journey, also I'm gonna be uploading a highlight video of that probably tomorrow. It took so long to edit 2 hours of stream into less than 30 minutes, but we have accomplished that. So yeah, I'll be uploading that tomorrow. But in that stream, I talked a bit about how one of my grad school rotations was working with Mycobacterium tuberculosis, and my goodness, that was the most boring project I ever worked on. I don't know. I don't know how someone can be a tuberculosis researcher. It's so much waiting. It's waiting forever. It cultures so slowly. Anyway, the bacterium that causes tuberculosis can be detected in specimens based on the presence of acid-fast bacilli. Tuberculosis infected immense portions of the global population throughout the 19th and 20th centuries, and much of the work of early microbiologists and other researchers was focused on the disease. Any TB, people with TB vaccinations as a kid? That's common if you weren't born in America, but I have my TB vaccination. It's really annoying. It causes problems because whenever I have to when you go to work sometimes in hospitals or places where there's vulnerable people, sometimes you have to like get a negative screening of tuberculosis, and my vaccination sometimes still causes like a false positive. I'm like, no, I'm just vaccinated, you guys. And then sometimes they're like, oh, you have to go get a chest screening to make sure you actually don't have tuberculosis. And I'm like, oh, please listen to me. It's stupid. But I also understand why they do it, but it always causes extra annoyance for me. It's like happened twice now. Maybe 3 times? No, twice. Ruth Ella Moore, a prominent researcher who is known for work on blood typing, dental cavities, and gut microbiome. What a triple threat. I worked on blood typing, dental cavities, and the gut microbiome. Yeah. As well as being the first Black woman to earn a natural science PhD, wrote her dissertation on new methods of isolating M. tuberculosis in order to better study it and develop a cure. These isolates would be critical in diagnosis and other medical procedures as well. What a career. Often a smear is prepared from a sample of this patient's sputum. That's the bleh, the kind of like the phlegm, but not really. Because I guess phlegm is more like other nastiness. And then stain using the Zeal-Nielsen technique. If acid-fast bacteria are confirmed, they are generally cultured to make a positive identification. Variations of this approach can be used as a first step in determining whether M. tuberculosis or other acid-fast bacteria are present, though samples from elsewhere in the body, such as urine, may contain other mycobacterium species. An alternative approach for determining the presence of M. tuberculosis is immunofluorescence. In this technique, fluorochrome-labeled antibodies bind to M. tuberculosis if present. Antibody-specific fluorescent dyes can then be used to view the mycobacterium with a fluorescence microscope. It's so funny looking. Actually, I wouldn't even call this funny looking. It's just really pretty. It's also kind of scary. I feel like this is like this would be used to like scare somebody, like in like a science, like a horror film that has like a sciencey theme, but not real science. You know, they would like call this like a zombie virus, and this is what the zombie researcher guy would be looking at under the microscope in the beginning of the movie, and go, and it's like he's in a poorly lit lab, and he's like looking at this growing, yeah. Okay, an ad break is starting also. I'm gonna use this time just to keep looking at this mycobacterium. And also drink water. And also drink my milk tea. I'm doing the classic, a drink for hydration and a drink for fun. Zeal-Nielsen staining has rendered these mycobacterium tuberculosis cells red and the surrounding growth indicator medium blue. Medium blue is a very funny word. Funny like way to describe it. Oh no. They mean growth indicator medium word blue. Not medium blue. I don't know why my brain thought they were describing the color as medium blue. Oh, I see. Capsule staining. Certain bacteria and yeast have a protective outer structure called a capsule. Since the presence of a capsule is directly related to a microbe's virulence, it's a hard word for me to pronounce. I suck at pronouncing words. If you have not been able to tell already, my pronunciation is not very good. Some have called it endearing, others have called it unprofessional. So, you know, take that for what it is. The ability to determine whether cells in a sample have capsules is an important diagnostic tool. Capsules do not absorb most basic dyes, therefore, a negative staining technique, staining around the cells, is typically used for capsule staining. The dye stains the background but does not penetrate the capsules, which appear like halos around the borders of the cell. The specimen does not need to be heat fixed prior to negative staining. Ooh, no heat fixing, nothing. Just a stain. One common negative staining technique for identifying encapsulated yeast and bacteria is to add a few drops of Indian ink or nigrosin to a specimen. Other capsular stains can also be used to negatively stain encapsulated cells. Alternatively, positive and negative staining techniques can be combined to visualize capsules. The positive stain colors the body of the cell, and the negative stain colors the background but not the capsule, leaving halo around each cell. Yeah, I kind of like. I think capsule staining is so pretty. I really like. I really like the color. Like the color, like the blurriness, you know. This one especially. What is this? Bacillus. Yeah, I figured it was like a bacillus, some kind of bacillus microbe. It's just so nice. Like it has such a pretty color. Like you're gonna tell me this, what a beautiful bacteria. Hi Resta. Hi, I hope you had a good stream. And then endospore staining. Endospores are structures produced within certain bacterial cells that allow them to survive harsh conditions. Gram staining alone cannot be used to visualize endospores, which appear clear when gram-stained cells are viewed. Endospore staining uses two stains to differentiate endospores from the rest of the cell. They are pretty, aren't they? I really like the colors of them. I think it's kind of funny just how pretty these pictures get, like microscope pictures can be, just simply because of the dyes we use. It's crazy what we can visualize. Science is just kind of amazing. Washing with water decolorizes the cell, but the endospore retains the green stain. The cell is then counterstained pink with safranin. Safranin. The resulting image reveals the shape and location of endospores if they are present. The green endospores will appear either within the pink vegetative cells or as separate from the pink cells together. If no endospores are present, then only the pink vegetative cells will be visible. Okay, I will be honest, I am not super familiar with like endospore staining or what the use case for this is, or understanding why you would really care about this, but I am gonna guess they'll go over this, and this is where we will learn together. I'm gonna guess this is like for clinical purposes. Like, surely it's because, like, for example, what you're seeing in this picture is bacillus, and that's a pretty clinically relevant bacteria. Oh yeah, see. Endospore staining techniques are important for identifying Bacillus and Clostridium, two genera of endospore-producing bacteria that only contain clinically significant species. Among others, B. anthracis, which causes anthrax, has been of particular interest because of certain concerns that its spores could be used as bioterrorism agent. Oh yeah, anthrax. Be careful of anthrax. Yeah. C. difficile is also a particularly important species responsible for the typically hospital-acquired infection known as C. diff. I wonder if malachite green has the mineral malachite in it, or if it's just named after its green color. I also wonder that too. I imagine it surely has. Maybe it does. I wonder if I can Google. I can Google this. Malachite green stain. Does it have malachite? Oh, despite its name, the dye is not prepared from the mineral malachite. The name just comes from the similarity of color. Ah, okay. Well, that answers that. That was no mineral. Not a single mineral is inside there. Just green. Flagella staining. Flagella, singular, flagellum, are tail-like cellular structures used for locomotion by some bacteria, archaea, and eukaryotes. Because they are so thin, flagella typically not be seen under a light microscope without a specialized flagella staining technique. Flagella staining thickens the flagella by first applying mordant, generally tannic acid, but sometimes potassium alum, which coats the flagella. Then the specimen is stained with, oh goodness, will I be able to pronounce this? Pararosanilline, most commonly, or basic fusion. I've never done flagella staining. I love flagella though. Wiggle, like, I just think, okay, it's like a fun word. It's also a fun structure, and it's just cool. I never worked in a lab where I needed to actually visualize it though, but I didn't know that you actually can't see it very easily under a microscope without like staining. Yeah, it's really fun. Yeah, flagella. How can you not like? You just want an exclamation point. Flagella, flagella. So you're telling me I might have like these guys that I've looked at under microscopes probably all had flagella, or not all, and I just didn't see it because they weren't stained with a specific thing. I guess they're really really thin. Like if a bacteria is already really thin, then can you imagine like how thin the flagella is and how specialized it needs to be to actually visualize it? I guess that makes sense. Though flagella staining is uncommon in clinical settings, the technique is commonly used by microbiologists since the location and number of flagella can be useful in classifying and identifying bacteria in a sample. When using this technique, it is important to handle the specimen with great care. Flagella are delicate structures that can easily be damaged or pulled off, compromising attempts to accurately locate and count the number of flagella. That is true. I guess that is true. It'd probably be like you'd have to be so careful if you actually care about flagella. You're probably, and I feel like they probably rip off just like that. Ow! Don't rip off their flagella, guys. That hurts. Simple stains. Okay, so this is just a review of what we went over. So we went over the basic stains, the acidic stains, the negative stains, the gram stain, the acid fast stain, the endospore stain, the flagella stain, and the hey, this ordering is messed up. The flagella stain was last, guys. It's not this way. I think my favorite was still the acid fast one, but I think that's simply because I'm biased. This is the one I remember distinctly working with when I was in my lab. I say distinctly working with, but apparently I didn't remember that you even had to heat fix it. Oh, I forgot this is about microscopes. I was like, why are we talking about microscopes again? But they had to bring it back to the microscope. Preparing specimens for electron microscopy. Samples to be analyzed using a TEM must have very thin sections. Very thin. Very thin. But cells are too soft to cut thinly, even with diamond knives. To cut cells without damage, the cells must be embedded in plastic resin and then dehydrated through a series of soaks in ethanol solutions. 50%, 60%, 70%. I kind of remember this. Kinda. Oh, you used to do gram stains? I was thinking, like, did I ever do gram stains? And I was like, I did do gram stains. I did. I feel like it's all blur now. Like, my experimental science days all feel blurred together, especially when I think about bacterial related stuff. Even though I love microbiology and bacteria, my work with them feels long ago now. Like my wet bench work. The ethanol just replaces the water in the cells, and then the resin dissolves in ethanol and enters the cell where it solidifies. Next, thin sections are cut using a specialized device using an okay, what? I thought I knew where this is going, but then they talked about an Ike Ultra microtome, and I don't know where we're going anymore. Mood, yeah. I feel like The dry lab gang. I don't think I would ever want to go back to wet lab. Wet lab is just it's way too tiring. Using my brain with like science is already like exhausting, but to use your brain and then also have to physically do activities and stand up and take care of things every day in a lab is not for me. Not for me. Not my cup of tea. Finally, samples are fixed to fine copper wire or carbon fiber grids and stained not with colored dyes but with substances like uranyl acetate or osmium tetroxide, which contain electron dense heavy metal atoms. Okay. Yeah, I've never done this. And I don't know what they're talking about. It's kind of fascinating how there's just so many different techniques out there in science. It's like no wonder science has to be very collaborative and like you have to talk to a lot of people because there's no way you would know how all these different techniques that exist out there. As ultra microtome used to prepare specimens for a TEM. A technician uses an ultra microtome to slice a specimen into thin sections. When samples are prepared for viewing using an SEM, they must also be dehydrated using an ethanol series. However, they must be even drier than is necessary for a TEM. Critical point drying with inert liquid carbon dioxide under pressure is used to displace the water from the specimen. After drying, the specimens are sputter coated with metal by knocking atoms off a palladium target with energetic particles. Sputtering coats prevents specimens from becoming charged by the SEM's electron. As that paragraph, I can't see it. As that paragraph kept going, I just kept getting more and more like, what the frick? This I am so this is so beyond what I've ever done. You submitted a sample for TEM before? Oh, that's so cool. This is like so far removed from what anything I ever did. So they're talking about like displacing atoms and sputter coating and electron beams. I understand it in theory, but I just yeah, this isn't not something I've ever done. I feel like I was much more situated in the simple biological sciences. Microconnections. Using microscopy to diagnose syphilis. More clinical relevance. The causative agent of syphilis is Treponema palladium, a flexible spiral cell that can be very thin, less than 0.15 micrometers, and match the refractive index of the medium, making it difficult to view using bright field microscopy. I like them too. I love the spiralies. They're so cool. I was very torn between like, do I want to have like more spirally stuff in my design? And then I realized it's like kind of it was not as fun to rig. It would be not that fun to rig the spirally. So I went with a more simple, just kind of like a wiggly thing, not spirally. Additionally, this species has not been successfully cultured in laboratory on an artificial medium. Therefore, diagnosis depends upon successful identification using microscopic techniques and serology. Serology is the analysis of body fluids, often looking for antibodies to a pathogen. Since fixation and staining would kill the cells, dark field microscopy is typically used for observing live specimens and viewing their movements. However, other approaches can also be used. For example, the cells can be thickened with silver particles and observed using a light microscope. It is also possible to use fluorescence or electron microscopy to view treponema. Look at them, they're all wiggly. This is with a dark field microscope, and then this is with a bright field microscope with a modified Steiner silver stain, which is used to visualize T palladium spirogets. Though the stain kills the cells, it increases the contrast to make them more visible. So I feel like it's kind of hard to see them in the bright field one. While not used for standard diagnostic testing, T. palladium can also be examined using the scanning electron microscopy. Ooh, look how wiggly it looks, how curly it is. It's really cool. It's crazy we can visualize it like with such graphics and resolution. Look at like A, which is a very 2D flat picture of it, and then can you believe we can do both A and C? In clinical settings, indirect immunofluorescence is often used to identify treponema. A primary unstained antibody attaches directly to the pathogen surface, and secondary antibodies tagged with a fluorescent stain attach to the primary antibody. Multiple secondary antibodies can attach to each primary antibody, amplifying the amount of stain attached to each treponema cell, making them easier to spot. The classic immunofluorescence, how it works. Just antibodies on top of antibodies on top of antibodies. Preparation and staining for other microscopes. Samples for fluorescence and confocal microscopy are prepared similarly to samples for light microscopy, except that the dyes are fluorochromes, and stains are often diluted in liquid before applying to the slide. Some dyes attach to an antibody to stain specific proteins on specific types of cells. Immunofluorescence. Others may attach to DNA molecules in a process called fluorescence in situ hybridization. Fish! I know fish! Fish is really popular in cancer biology. You use this a lot to confirm protein expression for certain kinds of cancer for diagnostics. Like you would look for a certain level of fish amplification ratio for the protein ERB2 for like breast cancer. So if you have a lot of this ERB2 protein, it will get amplified a lot, and you'll be able to visualize it through fluorescence in situ hybridization or fish. And that's usually a good way to say like, oh, you might have breast cancer, and you might be eligible for certain therapeutics depending on how much amplification of this ERB2 molecule you have. Yeah, it does remind, like, it's very similar to like ELISAs. There's always, I think there's so much of this like in any kind of immunofluorescence where it's like thing we want to look at, primary antibody, secondary antibody, and then the washing steps and all that, and visualization. I feel like if you know one immunofluorescence, you know like how to do all the immunofluorescence. Sample preparation for 2-photon microscopy is similar to fluorescence microscopy except for the use of infrared dyes. I don't know anything about 2-photon microscopy. Another thing that's like completely out of my depth of what scientific experience I got. Specimens for STM need to be on very clean and atomically smooth. Something about that word really trips me up. I want to say automatically smooth. Atomically smooth surface. They are often mica coated with gold 111. Toluene vapor is a common fixative. Wow. Again, I'm just kind of impressed. I always think the chemistry things that have the scientific techniques that are much more closely aligned with chemistry, and so you have to be more chemical focus, is always like interesting to me. Oh, the resolution to Cindy. From the results of the Gram stain, the technician now knows that Cindy's infection is caused by the spherical gram-positive bacteria that form grape-like clusters, which is typical of staphylococcal bacteria. It's staph. She has a staph infection, you guys. Don't run. This is like a reminder. Don't run around in camp because you're gonna fall and you're gonna get a staph infection on your knee. After some additional testing, the technician determines that these bacteria are the medically important species known as Staphylococcus aureus, a common culprit in wound infections. Because some strains of S. aureus are resistant to many antibiotics, skin infections may spread to other areas of the body and become serious, sometimes even resulting in amputations or death if the correct antibiotics are not used. Oh, will Cindy get will she be amputated? After testing several antibiotics, the lab is able to identify one that is effective against this particular strain of S. aureus. Cindy's doctor quickly prescribes the medication and emphasizes the importance of taking the entire course of antibiotics, even if the infection appears to clear up before the last scheduled dose. This reduces the risk that any especially resistant bacteria could survive, causing a second infection or spreading to another person. Okay, she didn't have her leg amputated. That's okay. That's good. That's good. Happy ending. This is a wait in the first chapter. We had a very unhappy ending for this clinical focus section of the chapter. One of the case studies they looked at, she was complaining about headaches and loss of memory and being thirsty constantly, and then it turns out she had a prion disease and she died. She had yeah, it was just really sad. It went like 0 to 100 really fast for that clinical focus. I was like, "Oh my gosh, she had a headache and she couldn't remember things and she and what do you mean she had a prion disease just like that?" Poor woman. I don't know if like these are real or fake stories, but regardless, I felt bad for her, and I'm glad that Cindy did not have the same fate. I was worried that this was gonna elevate all the way to, oh, we didn't catch her infection in time, and now her leg has to be amputated. It's okay. She just has to take her antibiotics. Take your antibiotics, guys. Do not not take your antibiotics. When you are prescribed the antibiotics, you have to take every single one of those pills they prescribe you. It's crazy how that feels like common knowledge, but I know so many people personally that have said like, "Oh yeah, like I usually stop taking them when I realize like everything's done." And I'm like, "No, you're not supposed to do that. You're not supposed to do that. Don't you listen to your doctor? Your doctor said don't do that. And why don't you listen? Why can't you just be normal and take all your antibiotics, guys?" Eyes on ethics. Ooh, I like their last section on ethics. Microscopy and antibiotic resistance. As the use of antibiotics has proliferated in medicine as well as agriculture, microbes have evolved to become more resistant. Like me, I'm a very resistant microbe. Strains of bacteria such as methicillin-resistant S. aureus, MRSA, which has developed a high level of resistance to many antibiotics, are an increasingly worrying problem. So much so that research is underway to develop new and more diversified antibiotics. Fluorescence microscopy can be useful in testing the effectiveness of new antibiotics against resistant strains like MRSA. In a test of one new antibiotic derived from a marine bacterium, MC21A bromophene, researchers used the fluorescent dye cytoxgreen to stain samples of MRSA. Cytoxgreen is often used to distinguish dead cells from living cells with fluorescence microscopy. Live cells will not absorb the dye, but cells killed by an antibiotic will absorb the dye, since the antibiotic has damaged the bacterial cell membrane. Oh, that makes a lot of sense how that... I've heard of all this before. I just didn't actually understand why, the mechanism of why it does this. That seems to be like my common pitfall in my scientific knowledge, is I know something, but I don't actually understand why it works the way it is. I think it's not usual for the why to be taught, or at least for me not to be remembered. In this particular case, MRSA bacteria that had been exposed to MC21A did indeed appear green under the fluorescence microscope, leading researchers to conclude that it is an effective antibiotic against MRSA. Of course, some argue that developing new antibiotics will only lead to even more antibiotic-resistant microbes, so-called superbugs that can spawn epidemics before new treatments can be developed. For this reason, many health professionals are beginning to exercise more discretion in prescribing antibiotics. I feel like that's true in theory, but I think a lot of health professionals still tend to overprescribe antibiotics. Whereas antibiotics were once routinely prescribed for common illnesses without a definite diagnosis, doctors and hospitals are much more likely to conduct additional testing to determine whether an antibiotic is necessary and appropriate before prescribing. A sick patient might reasonably object to the stingy approach to prescribing antibiotics. To the patient who simply wants to feel better as quickly as possible, the potential benefits of taking an antibiotic may seem to outweigh any immediate health risks that might occur if the antibiotic is ineffective. But at what point do the risks of widespread antibiotic use supersede the desire to use them in individual cases? Yeah, it's definitely like a difficult thing to balance. So many antibiotics are used by factory farms too. Yeah, it's just like it's not even like you said. It's not even just in a clinical setting. It's just overused wherever, whatever field can use antibiotics, they're overusing it, and it's causing problems to the other fields. It's causing problems just to everyone. We gotta lay off on this antibiotic like usage, but it's like they said, it's really hard. I'm not sure. I'm sure in terms of like factories and like farm factories, it's more like probably like cost efficiency. It's probably very cheap to just like willy nilly do all this. But like maybe it's like only short term cheap though. Like surely in the long term, it's going to cause problems and end up being more expensive when they have to like figure out how to solve the issues of antibiotic resistance. Oh, we finished the chapter. We're finished with micros. We're not we're not doing questions. I'm not doing any questions. Oh, we're done. We're done with the microscopes. We're free from the microscopes. We can get into some actual interesting like biology things again. Yippee! Yay, yay, yay! Yeah! The cell. Oh, that actually sounds interesting. And look, there's pretty pictures. There's pretty pictures that lure us in. Microorganisms vary visually in their size, shape, and can be observed microscopically, but they can also vary in invisible ways, such as their metabolic capabilities. Yeah, I really, they use this picture. Yeah, yeah, it does have a spooky face. Yeah, quote unquote face. I love all of these pictures. Wow, how beautiful. Wow, the microscopic world is just so pretty. It's just insane how pretty and colorful it is. I know a lot of the colors are simply stain. Like we're doing that. It's a bit of like a human interference aspect to it, but isn't it like kind of like art in a way? It's it's just so like lively. It's fascinating how it's in nature just like that. Life takes many forms, from giant redwood trees towering hundreds of feet in the air to the tiniest known microbes. Me, I am the tiniest known microbe, which measure only a few billionths of a meter. Humans have long pondered life's origin and debated the defining characteristics of life, but our understanding of these concepts has changed radically since the invention of the microscope. In the 17th century, observations of microscopic life led to the development of the cell theory, the idea that the fundamental unit of life is the cell, that all organisms contain at least one cell, and that cells only come from other cells. The classic theory. Like, I think this is like the first lesson you usually learn in Biology 101. At least I distinctly remember going into my first biology class, and it was like the cell. Day one, the cell. And then you just learn about cell theory and all that. Despite sharing certain characteristics, cells may vary significantly. The two main types of cells are prokaryotic cells, lacking a nucleus, and eukaryotic cells, containing a well organized membrane bound nucleus. Each type of cell exhibits remarkable variety in structure, function, and metabolic activity. This chapter will focus on the historical discoveries that have shaped our current understanding of microbes, including their origins and their role in human disease. We will then explore the distinguishing structures found in prokaryotic and eukaryotic cells. Ah, history lessons. I'm not, no joke, no joke. I think the history lesson parts of these, like, This textbook has been really interesting so far. I am not super well versed in biological history, so I've appreciated learning how certain discoveries came to be. The spontaneous generation. By the end of this section, we'll be able to explain the theory of spontaneous generation and why people once accepted it as an explanation for the existence of certain types of organisms. Explain how certain individuals, Van Helmont, Reddy, Needham, Spallanzani, and Pasteur, tried to prove or disprove spontaneous generation. All right, I read this as like Generation Alpha, but like the spontaneous generation. No, they're literally talking about things coming out of spawning from nothing. Let's see what our clinical focus. What's our clinical focus for this chapter? Will this have a sad ending? Any guessers? Part 1. Barbara is a 19-year-old college student living in the dormitory. In January, she came down with a sore throat, headache, mild fever, chills, and a violent but unproductive, i.e. no mucus cough. To treat these symptoms, Barbara began taking an over-the-counter cold medication, which did not seem to work. Sad. In fact, over the next few days, while some of Barbara's symptoms began to resolve, her cough and fever persisted, and she felt very tired and weak. Very relatable, Barbara. I think I was feeling like this a few weeks ago. It's like the worst. What type of respiratory diseases may be possible? Is she going to be okay? This sounds innocuous. Humans have been asking for millennia. Where does new life come from? Religion, philosophy, and science have all wrestled with this question. One of the oldest explanations was the theory of spontaneous generation, which can be traced back to the ancient Greeks and was widely accepted through the Middle Ages. Wow, I didn't realize they believed things just spawned like that for so long. The theory of spontaneous generation. The Greek philosopher Aristotle, 384 to 322 BC, was one of the earliest recorded scholars to articulate the theory of spontaneous generation, the notion that life can arise from non-living matter. I don't know about that one, Aristotle. Yeah, I don't know about that one. Aristotle proposed that life arose from non-living material if the material contained pneuma, spirit or breath, as I guess like pneumonia, pneuma. As evidence, he noted several instances of the appearance of animals from environments previously devoid of such animals, such as the seemingly sudden appearance of fish in a new puddle of water. That is so funny to think about. He just like stared at some water and said, "Look, this fish just spawned." That's it. Spontaneous generation. A fish out of nowhere. But like, was he even looking? How did he know there was nothing there? I feel like he did not do a good job like conducting a basis for his hypothesis here. I feel like he just simply looked at a puddle of water and said, "Yeah, there's no fish there." And then bam, fish, and that was all it took for him to think that this theory was sound. This theory persisted into the 17th century, and it carried to the 17th century when scientists undertook additional experimentation to support or disprove it. You're telling me everyone was like, "All right, bet, things do spawn." When we humans, we just spawn in the tummy. By this time, the proponents of the theory cited how frogs simply seemed to appear along the muddy banks of the Nile River in Egypt during the annual flooding. Others observed that mice simply appeared among grain stored in barns with thatched roofs. I'm being hard on them, but that's all they had. Well, it's not like they had much to think about. They simply observed, and that's what they thought. Others, when the roof leaked and the grain molded, mice appeared. Whoa, the mice just spawned. They spawned into my farm. How could they do that? John Baptista van Helmont, a 17th century Flemish scientist, proposed that mice could arise from rags and from rags and wheat kernels left in an open container for 3 weeks. In reality, such habitats provided ideal food sources and shelter for mouse populations to flourish. It's not the most sound experiment. You would think. Again, I'm being too hard on them. There's none of this rigor like that I'm thinking about existed back then. You just leave some rags and some wheat kernels, and you go, that's my science experiment. One of Van Helmont's contemporaries, Italian physician Francesco Redi of 1626 to 1697, performed an experiment in 1668 that was one of the first to refute the idea that maggots, the larvae of flies, spontaneously generate on meat left out in the open air. I really think spontaneous. I think the word spontaneous generation is just so funny to me because I'm imagining it in like a video game where something just literally spawns like in the middle of nowhere. That like literally spontaneous generation, and it's like so funny to think about how people of back then were like, yeah, things are just spawning out of nowhere, you guys. It's insane. Animals, they just spawn. If you don't look away, they're gonna spawn. Reddy left meat in each of 6 containers. Oh, okay. This guy, he was conducting a real science experiment here, like with controls and everything. 2 were open to the air, 2 were covered with gauze, and 2 were tightly sealed. His hypothesis was supported when maggots developed in the uncovered jars, but no maggots appeared in either the gauze-covered or tightly sealed jars. He concluded that maggots could only form when flies were allowed to lay eggs in the meat, and that the maggots were the offspring of flies, not the product of spontaneous generation. What? What? That's crazy. Yeah, and there's his science experiment. Open container, formation of maggots. No. It's kind of gross. It's kind of nasty. In 1745, John Needham, 1713 to 1781, published a report of his own experiments in which he briefly boiled broth infused with plant or animal matter, hoping to kill all pre-existing microbes. He then oh wow, what? Okay, now that's interesting. He really developed an interesting protocol here. He then sealed the flask. After a few days, Needham observed that the broth had become cloudy and a single drop contained numerous microscopic creatures. He argued that the new microbes must have arisen spontaneously. So close. He was just like so close to like getting there. That might be the most frustrating of science experiments they've described. Where someone is just like he was really on the right track. Wrong conclusion. Completely wrong conclusion. But you were close, buddy. You almost had it. In reality, however, he likely did not boil the broth enough to kill all pre-existing microbes. And yeah, like, how would he have known? Can you even blame him for coming to that wrong conclusion? Because you used what you assumed, which is kind of correct, that microbes will die if they are heated up a certain temperature, but it's not like you understand exactly what temperature they need to be heated up at. Not at this point, I don't think. Lazzaro Spallanzani, 1729 to 1799, did not agree with Needham's conclusion. Oh, the girls are fighting. The girls are fighting. However, and performed hundreds. He said, no, girl, I am going to outscience you. Performed hundreds of carefully executed experiments using heated broth. As in Needham's experiment, broth in sealed jars and unsealed jars was infused with plant and animal matter. Spallanzani's experiments results contradicted the findings of Needham. Heated but sealed flasks remained clear without any signs of spontaneous growth, unless the flasks were subsequently opened to the air. This suggested that microbes were introduced into these flasks from the air. They did it. He found the connection. In response to Spallanzani's findings, Needham argued that life originates from a life force that was destroyed during Spallanzani's extended boiling. Any subsequent sealing of the flask then prevented new life force from entering and causing spontaneous generation. Oh, he's making sense. He's making sense. He kind of just like owned all of them. Francesco Redi. Beautiful locks of hair. Beautiful. John Needham. Not as beautiful. It's really giving... This is like George Washington-esque hair. And Lazzaro Spallanzani. This man, he was so heated. To do hundreds of experiments because you were upset at some guy's experiment, that's like true scientific passion. Disproving spontaneous generation. Well, didn't they just disprove it? I guess that probably was not enough to disprove it, huh? Like if a theory has been going on since Aristotle's, like you're not gonna like accept that it's disproven with just one guy's experiment, I'm guessing. The debate over spontaneous generation continued well into the 19th century, with scientists serving as proponents of both sides. To settle the debate, the Paris Academy of Sciences offered a prize for the resolution of the problem. Louis Pasteur. This guy is a super science OG. A prominent French chemist who had been studying microbial fermentation and the causes of wine spoilage accepted the challenge. In 1858, Pasteur filtered air through a gun cotton filter. A what? And upon microscopic examination of the cotton found it full of microorganisms, suggesting that the exposure of a broth to air was not introducing a life force to the broth, but rather airborne microorganisms. Is that what they thought? Did they really think like air had life force? I guess that's what Aristotle was saying. Later, Pasteur made a series of flasks with long twisted necks, swan neck flasks, in which he boiled broth to sterilize it. His design allowed air inside the flasks to be exchanged with air from the outside, but prevented the introduction of any airborne microorganisms, which would get caught in the twists and bends of the flask's neck. If a life force besides the airborne microorganisms were responsible for microbial growth within the sterilized flask, it would have access to the broth, whereas microorganisms would not. He correctly predicted that sterilized broth in his swan neck flask would remain sterile as long as the swan necks remained intact. However, should the necks be broken, microorganisms would be introduced, contaminating the flask and allowing microbial growth within the broth. Wow. Okay, I feel like I was giving some of the scientists a lot, but then Louis Pasteur came to the scene and said, "This is how you do science." And yeah, it's pretty impressive how he was able to really put together such an elegant science experiment. Pasteur's set of experiments irrefutably disproved the theory of spontaneous generation and earned him the prestigious Al-Humbert Prize from the Paris Academy of Sciences in 1862. In a subsequent lecture in 1864, Pasteur articulated Om vivum ex vivo. Life only comes from life. Damn. What a phrase. In this lecture, Pasteur recounted his famous swan neck flask experiment, stating that life is a germ and a germ is life. Never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. Wow. He stepped on their necks. He said, "No, girls, you were all wrong." He's kind of cool. He's kind of sick with it. To Pasteur's credit, it never has. Damn, microbiology textbook writers. That was also a sick line. You know, I'm kind of a Louis Pasteur's stan now. He kind of stepped on the science necks from everyone from the below the 17th century. Wow. That's him, right? You know, to his I feel like he's not giving himself enough credit. It is a simple experiment in theory, but it's so like it's pretty elegant. It's pretty elegant, you know. Because I'm trying to imagine myself as a Mr. 17th century scientist. Would I think this deeply about it? Probably not. I mean, I feel like I have all the knowledge I have now, and I would have a hard time coming up with an experiment for for something relevant in today's scientific field right now. I know it's like hard to compare, but damn. So cool. Foundations of modern cell theory, learning objectives. By the end of this section, we will be able to explain the key points of cell theory and the individual contributions of Hooke, Schleiden, Schwann, Remak, and Virchow. Oh my gosh, the European names just don't stop. Explain the key points of endosymbiotic theory and cite the evidence that supports this concept. Explain the contribution of Semmelweis, Snow, Pasteur, Lister, and Koch to the development of germ theory. Ooh, germ theory. While some scientists were arguing over the theory of spontaneous generation, other scientists were making discoveries leading to a better understanding of what we now call the cell theory. Modern cell theory has two basic tenets. All cells come from other cells, the principles of biogenesis, and cells are the fundamental units of organisms. Today, these tenets are fundamental to our understanding of life on Earth. However, modern cell theory grew out of the collective work of many scientists. That's funny. All this bickering about what spontaneous generation is like happening in the background of this. But it sounds like if this tenet has already been sprung, don't they kind of understand like spontaneous generation can't possibly be real if we know all cells come from other cells? No? Yeah? The origins of cell theory. The English scientist Robert Hooke first used the term cells in 1665 to describe the small chambers within cork that he observed under a microscope of his own design. We learned about this in chapter 1, I remember this. To Hooke, thin sections of cork resembled honeycomb or small boxes or bladders of air. He noted that each cavern, bubble, or cell was distinct from the others. At the time, Hooke was not aware that cork cells were long dead and therefore lacked the internal structure found within living cells. Look at him. He's looking at cells. I guess, like, it's funny that the first observation of cells comes from corks. That would not be, like, what I would have thought. But also, maybe there's nothing... What else would they be looking at? I have no idea. I would have thought maybe something human. Despite Hooke's early description of cells, their significance as the fundamental unit of life was not yet recognized. But that makes sense why it wouldn't be recognized. Who's gonna care about, like, who's gonna take what you saw from a cork seriously and apply that to biological relevance? They're probably not even thinking about what corks are made out of. I'll tell you, I feel like you ask someone what a cork is made out of now, and do you think people would know? I have a feeling if I walked up to a random person and they asked them, do you know what a cork is made out of? They wouldn't know. So, you know, I kind of get this in modern day. Nearly 200 years later, in 1838, Matthias Schleiden, 1804 to 1881, a German botanist who made extensive microscopic observations of plant tissues, described them as being composed of cells. Visualizing plant cells was relatively easy because plant cells are clearly separated by their thick cell walls. Oh, you know, I didn't think of that about plant cells, but it makes perfect sense why a plant cell would be the first thing to be observed compared to human. Schleiden believed that cells form through crystallization rather than cell division. Close. Not quite, but close. Close one. Theodore Schwann. Is this Schwann cell guy? Surely this is the Schwann cell guy. 1810-1882, a noted German physiologist made similar microscopic observations of animal tissue. In 1839, after a conversation with Schleiden, Schwann realized that similarities existed between plant and animal tissues. This laid the foundation for his idea that cells are the fundamental components of plants and animals. Wow. What a... I'm sure they're simplifying what his thoughts were, but like that paragraph alone makes him sound like a genius. In the 1850s, two Polish scientists living in Germany pushed this idea further, culminating in what we recognize today as the modern cell theory. The modern cell theory. In 1852, Robert Remak, 1815 to 1865, a prominent neurologist and embryologist, published convincing evidence that cells are derived from other cells as a result of cell division. You know, makes sense. However, this idea was questioned by many in the scientific community, and they did not believe him. And why should they? 3 years later, Rudolf Virchow, 1821 to 1902, whoa, the fact we're in the 19th century now feels like whoa, we've made a lot of progress. A well-respected pathologist published an editorial essay entitled Cellular Pathology, which popularized the concept of cell theory using the Latin phrase omnis cellula a cellula, all cells arise from cells, which is essentially the second tenet of modern cell theory. Given the similarity of Virchow's work to Remak's, there is some controversy as to which scientist should receive credit for articulating cell theory. See the following Eye on Ethics feature for more about this controversy. Okay. Oh, plagiarism. Science and plagiarism. This is kind of juicy. I don't know how juicy they're going to get here, but there is some like controversial plagiarism in science history. Rudolf Virchow, a prominent Polish-born German scientist, often remembered as the father of pathology. Well known for innovative approaches, he was one of the first to determine the causes of various diseases by examining their effects on tissues and organs. He was also among the first to use animals in his research, and as a result of his work, he was the first to name numerous diseases and created many other medical terms. Over the course of his career, he published more than 2,000 papers. 2,000! I think in my I don't think I'm even on track to get 100. If that. 2,000. 2,000. 2. Not 2. 200. 2,000. But also, wasn't publishing papers easier back then? Maybe I'm coping. And headed various important medical facilities, including Charité, University. I'm sorry, I don't know how to pronounce that. University of Berlin is the Medical University of Berlin is what I'm going to assume that translate to. A prominent Berlin hospital and medical school. Oh, I'm so good. I'm so good at languages. But he is perhaps best remembered for his 1855 editorial essay titled Cellular Pathology, published in Archive for Pathologists Anatomy, the Archive of Pathology, Anatomy, and Physiology. Yeah, you see that? I'm pretty good with languages. A journal that Virchow himself co-founded and still exists today. Despite his significant scientific legacy, there is more controversy regarding this essay in which Virchow proposed the central tenet of modern cell theory that all cells arise from other cells. Robert Remak, a former colleague, they were colleagues? They were colleagues? I thought this was gonna go into they don't even know each other, but they seem to have come across the same information. Who worked in the same laboratory as Virchow in the University of Berlin had published the same idea 3 years before. Though it appears Virchow was familiar with Remak's work, he neglected to credit Remak's ideas in his essay. The nerve! The nerve! When Remak wrote a letter to Virchow pointing out his similarities between his ideas and his own, Virchow was dismissive. In 1858, in the preface to one of his books, Virchow wrote that his 1855 publication was just an editorial piece, not a scientific paper, thus there was no need to cite Remark's work. What a... He is so rude. Ooh, I don't like this man. I don't like this man one bit. By today's standards, Virchow's editorial piece would certainly be considered an act of plagiarism. Yeah, it would. Yeah, it would. Oh my god, to be dismissive to your colleague and your colleague just going, "Hey, buddy, you kind of took my idea. You want to?" He didn't even say, "Hey, you should like revoke that." He was just saying, "Hey, buddy, can I get some credit? Can I get a little like?" Can I get something? Can you put like name drop me? Cause you kind of stole my idea. It's just saying, but just credit me. It's all good. He said this isn't even a scientific paper. It's not even a scientific paper. So I don't need to cite your work. Oh, ooh. However, since he presented Remark's idea as his own, yeah. However, in the 19th century, standards for academic integrity were much less clear. Ooh. Virchow's strong reputation, coupled with the fact that Remark was a Jew in a somewhat anti-Semitic political climate, geopolitics, creating problems in the scientific community, never changes, shielded him from any significant repercussions. Today, the process of peer review and the ease of access to the scientific literature help discourage plagiarism. Although scientists are still motivated to publish original ideas that advance scientific knowledge, those who would consider plagiarizing are well aware of the serious consequences. In academia, plagiarism represents the theft of both individual thought and research, an offense that can destroy reputations and careers. This is a bit... It's like... I do think it should be ending careers, but lately I feel like it's not really doing a whole lot, probably because it's just becoming too much of a widespread problem. It's actually so hard to catch fake papers now, or plagiarized papers, or fake papers, or paper mill papers, because there's just so much of it, and it's like partly to blame with AI, also partly to blame with the nature of science being a whole perish or publisher perish kind of like phenomenon. It's really bad. I really like this the website that tracks a bunch of controversial scientific publication issues, especially when it comes to things that are plagiarized or being redacted because there's also fake science. It is insane how much bad science is out there. It can be so difficult. This is a bit different of a ethics matter compared to just plagiarism alone. But academic integrity feels like it's at the lowest it's ever been. While also being maybe I want to say it's higher than back then in this 19th century example. But I don't know. It almost feels like things are just getting worse. They're just getting worse. There's so much slop papers out there. There are papers out there that are making fake Western bots. They're making fake graphs, fake figures. They are very obviously fake. Like, I've seen papers with Western bots. They literally just copy and paste lanes from one another to get the results they want to show that line up with what they're trying to present as the result of their paper. And it's insane that they were even accepted, because I look at them immediately and I go, yeah, that's not real. that's I know you want to pretend that is a real Western bot you're showing me, but that's not a real Western bot. Oh, even worse are flow cytometry results. Flow cytometry results that are just they just copy and paste. They copy and paste sections of the graph to other sections of the graph, and I'm like, that's not real. Or they don't even do that, and they just repeat the figure. That one's insane to me. They just repeat the figure into different parts of the paper. Like they'll repeat the control figure and then later paste that into like an experimental part, like an experimental figure, and then try to pass it off as like, yeah, that's how the experimental data looked. Like, no, no, it didn't. It's right there. And it gets published. It actually gets published. It's so insane. I went on a bit of a tangent. Man, I can't believe he just took his colleague's work. That's still crazy to me. Endosymbiotic theory. Well, as scientists were making progress toward understanding the roles of cells in plant and animal tissues, others were examining the structures within the cells themselves. In 1831, Scottish botanist Robert Brown, 1773 to 1858, I feel like this is a guy that lived longer than most of the people we've mentioned, was the first to describe observations of nuclei in which he observed in plant cells. Then, in the early 1880s, German botanist Andreas Schimper, 1856 to 1901, was the first to describe the chloroplasts of plant cells, identifying their role in starch formation during photosynthesis and noting that they divided independent of the nucleus. Based upon chloroplast's ability to reproduce independently, Russian botanist Konstantin Mershowski, 1855 to 1921. 1921! He made it to the roaring 20s! Suggested in 1905 that chloroplasts may have originated from ancestral photosynthetic bacteria living symbiotically inside a eukaryotic cell. He proposed a similar origin for the nucleus of plant cells. This was the first articulation of the endosymbiotic hypothesis and would explain how eukaryotic cells evolved from ancestral bacteria. Ooh. I think it's interesting how many of these hypotheses and theories started with plant cells, but like I said also earlier, it makes sense because of how much easier it is to visualize a plant cell. Mershowski's endosymbiotic hypothesis was furthered by American anatomist Ivan Wallin, 1883 to 1969. 1969. That's so recent. My mind got blown at how recent that is. Okay, it doesn't sound recent, but that's like people alive today are born at this time, who began to experimentally examine the similarities between mitochondria, chloroplasts, and bacteria. In other words, to put the endosymbiotic hypothesis to the test using objective investigation. Wallin published a series of papers in the 1920s supporting the endosymbiotic hypothesis, including a 1926 publication co-authored with Mirashkowski's. Wallin claimed he could culture mitochondria outside of their eukaryotic host cells. Many scientists dismissed his cultures of mitochondria as resulting from bacterial contamination. Oh, poor Wallin. They said, no, you suck. Modern genome sequencing work supports the dissenting scientists by showing that much of the genome of mitochondria had been transferred to the host cell's nucleus, preventing the mitochondria from being able to live on their own. Oh, well, yeah, to be fair, he did kind of suck. Okay, to be fair, he was not cooking anything. He was not onto anything. That's okay, buddy. You know, not all the scientists they listed here were hitters. They weren't always successful. Wallen's ideas regarding the endosymbiotic hypothesis were largely ignored for the next 50 years because scientists were unaware that these organelles contain their own DNA. However, with the discovery of mitochondrial and chloroplast DNA in the 1960s, the endosymbiotic hypothesis was resurrected. It was back. Lynn Margulis, 1938 to 2011. She was just alive. Just a little bit ago. Just a bit ago. Okay, that was like over a decade ago now. But that was just a bit ago. An American geneticist published her ideas regarding the endosymbiotic hypothesis of the origins of mitochondria and chloroplasts in 1967. In the decade leading up to her publication, Margulis reviewed the literature and argued that the eukaryotic organelles such as mitochondria and chloroplasts are of prokaryotic origin. She presented a growing body of microscopic genetic, molecular biology, fossil, and geological data to support her claims. Dang. What a paper. She had it all. She laid her table, like her hand to the table and said, "Yeah, I am right. Now look at all this evidence and data from all of these different aspects. I am super correct on this." I kind of love the scientists that are very, very confident that they are correct on a hypothesis. Again, this hypothesis was not initially popular, but mounting genetic evidence due to the advent of DNA sequencing supported the endosymbiotic theory, which is now defined as the theory that mitochondria and chloroplasts arose as a result of prokaryotic cells, establishing a symbiotic relationship within a eukaryotic host. With Margulis' initial endosymbiotic theory gaining wide acceptance, she expanded on the theory in her 1981 book Symbiosis in Cell Evolution. I read this book, or at least I know of this book, I think. I'm pretty sure this was recommended in my bio class. In it, she explains how endosymbiosis is a major driving factor in the evolution of organisms. More recent genetic sequencing and phylogenetic analysis show that mitochondrial DNA and chloroplast DNA are highly related to their bacterial counterparts, both in DNA sequence and chromosome structure. However, mitochondrial DNA and chloroplast DNA are reduced compared with nuclear DNA because many of the genes have moved from the organelles into the host cell's nucleus. Additionally, mitochondrial and chloroplast ribosomes are structurally similar to bacterial ribosomes rather than to the eukaryotic ribosomes of their hosts. Last, the binary fission of these organelles strongly resembles the binary fission of bacteria, as compared with mitosis performed by eukaryotic cells. Since Margulis' original proposal, scientists have observed several examples of bacterial endosymbionts in modern-day eukaryotic cells. Examples include the endosymbiotic bacteria found within the guts of certain insects such as cockroaches, and photosynthetic bacteria like organelles found in protists. I don't know. I'm not, again, the theoretical aspects of like why science is the way it is. I am sure I was taught all this. I'm no doubt I was taught all this. Just never has stuck in my brain, but it all like makes a bunch of sense. The endosymbiotic theory. Infoldings in the plasma membrane of an ancestral cell gave rise to endomembrane components, including a nucleus and endoplasmic reticulum. And then aerobiotic bacterium, photosynthetic bacterium, mitochondria. Oh, hello, Gren. Oh, you don't need to apologize. There's no need to apologize. It's not like there's any expectation for you to always be here or even to be here on time. The germ theory of disease. Prior to the discovery of microbes during the 17th century, other theories circulated about the origins of disease. Now that, I guess now this is more about this is more about, what the bacteria are doing. Germ theory. They've talked a lot now about why the cells exist and the bacteria exist. No spontaneous generation. They were able to disprove spontaneous generation. Things don't just randomly spawn in the air. So prior to the discovery of microbes during the 17th century, other theories circulated about the origins of disease. For example, the ancient Greeks proposed the miasma theory, which held that disease originated from particles emanating from decomposing matters, such as that in sewage or cesspits. That would make sense. You know, you can't even fault the ancient Greeks for thinking that, because they're not entirely wrong. They're, sewage bad. Sewage give disease. Yeah, they're right. They're right. Such particles infected humans in close proximity to the rotting material. Diseases including the Black Death, which ravaged Europe's population during the Middle Ages, were thought to have originated in this way. In the 11th century, Persian physician Ibn Sina, sometimes referred to as Avicenna, proposed that tuberculosis was likely to spread by people's breath when in close proximity. And, they're not wrong either. Tuberculosis was, in fact, it's more like they've got the right idea. The conclusion is just a bit wrong. They were just a little off with the conclusion, but also it's 11:55. Am I expecting them to understand what a mycobacterium tuberculosis is? It's in people's lungs and it's infecting people. It's not exactly their breath. No, no, no. Arab physician Ibn Zur, writing in about 1155, documented that the common skin condition scabies was caused by tiny mites that bored into the skin. Those scabies mites are about 0.5 millimeter long and therefore not technically microscopic, and their skin tunnels are often visible. Ibn Zur's discovery gave more evidence that unseen substances or creatures cause diseases. He's right. He's right. In 1546, Italian physician Girolamo, oh my gosh, this guy's name is so Italian, Fracastoro, proposed in his essay De Contagione et Contagiosis Morbis, the morb, that seed-like spores may be transferred between individuals through direct contact, exposure to contaminated clothing, or through the air. Oh, it's kind of crazy. 1546, he's kind of cooking. He kind of like saw it. Not the contagious morbis. We now recognize Fracastoro as an early proponent of the germ theory of disease, which states that diseases may result from microbial infection. However, in the 16th century, Fracastoro's ideas were not widely accepted and would be largely forgotten until the 19th century. Damn. He was cooking so hard with like the understanding of perhaps there's things we do not see that are causing diseases, and they ignored his ideas for 3 centuries. It's okay. I'll remember you, Italian physician Girolamo Fracastoro, and your contagious morb. 1847, Hungarian obstetrician Ignaz Semmelweis observed that people who gave birth in hospital wards staffed by physicians and medical students were more likely to suffer and die from puerperal fever after childbirth, 10 to 20% mortality rate, than were people in wards staffed by midwives, 1% mortality rate. That movie was very strange. Semmelweis observed medical students performing autopsies and subsequently carrying out vaginal examinations on living patients without washing their hands in between. Ew. Ew. Those were some. I guess it's not like they didn't have the proper procedures back then. I can't believe they were just not washing their hands. That's insane to think about. Nowadays, like, it's so. The amount of like washing and cleansing you have to do in between experiments or just doing anything in a hospital, it's kind of like, and the thought of they weren't even washing their hands. Not a single hand wash was done. He suspected that the students carried disease from the autopsies to the patients they examined. They went from autopsy to looking at helping people give birth. It was completely creamed in the 18th century. My goodness. His suspicions were supported by the untimely death of a friend, a physician who contracted a fatal wound infection after a post-mortem examination of a woman who had died of puerperal infection. The dead physician's wound had been caused by a scalpel used during the examination, and his subsequent illness and death closely paralleled that of the dead patient. Oh my gosh, okay. Not washing your hands is one thing. They took scalpels from an autopsy and then just dragged that that scalpel that was used on an infected patient and then examined other patients with it. Oh my god. Oh my god. It's crazy because I don't. Okay, 1847 sounds like a long time ago, and it is. It is a long time ago, but it's also not that long ago when we think about how in this book they've been mentioning things in like the BCs. So to think that just in 1847 they still didn't realize that they needed to use a different instrument and wash their hands. It just shows you like I feel like so much of our modern medical advancements happen in this like modern era, like of the era of where people are still alive to this day. Yeah, of course, in a geological aspect, it's very different. It is just a tiny blip. Whoa, his head is something else. His noggin is kind of crazy. Although Semmelweis did not know the true cause of puerperal fever, he proposed that physicians were somehow transferring the causative agent to their patients. He suggested that the number of puerperal fever cases could be reduced if physicians and medical students simply washed their hands with chlorinated lime water before examining every patient. He was right, he was cooking. When this practice was implemented, the maternal mortality rate in people cared for by physicians dropped to the 1%, same 1% mortality rate observed among people cared for by midwives. That's crazy. They like saw like an immediate effect. It went down all the way to from 20% to 1%. That's a huge drop. He does. His cranium is massive. I don't know if this is like I imagine this is a drawing. I feel like the artist kind of did him dirty. This demonstrated that hand washing was a very effective method for preventing disease transmission. Despite this great success, many discounted Semmelweis' work at the time, and physicians were slow to adopt the simple procedure of hand washing to prevent infections in their patients because it contradicted established norms for that time period. Oh my gosh. I feel like that whole thing I just read is still relevant to this day. People just not wanting to they get new information and they don't want to do that even though there's new information telling them, "Hey, this is really good for you." And they'll still go, "No, we do things this way and we've been doing things this way for so long now." Oh my gosh, humans just don't change. Even it's insane. It's so insane. Like I feel like this parallels so much with like the pandemic. We get information about how there's ways to prevent infection and people go, "Nah, I'm here to do what I've always done." Very crazy. Around the same time Semmelweis was promoting hand washing, man, even promoting hand washing is like it's a funny parallel. I can't believe we had to promote hand washing. In 1848, British physician John Snow conducted studies to track the source of cholera outbreaks in London. By tracing the outbreaks of two specific water sources, both of which were contaminated by sewage, Snow ultimately demonstrated that cholera bacteria were transmitted via drinking water. Snow's work is influential in that it represents the first known epidemiological study, and it resulted in the first known public health response to an epidemic. The first public health response to an epidemic. 1848. That's kind of crazy. The work of both Semmelweis and Snow clearly refuted the prevailing miasma theory of the day, showing that disease is not only transmitted through the air, but also through contaminated items. It's everywhere. Yeah. There was, like, I think most states had a huge problem with how they handled COVID. It's really, really unfortunate to see, but you can't even blame the people. I think there's some blame to how people responded, but it's also like up to governing agencies to do better, to educate better, to do a better response. Just everyone fail. Like, it's like a mix of like failure on so many different levels. Administrative failures, the governing agency failures, also like a federal failure. Like, I mean, yeah, you could blame it on the fact like the country failed everyone. Yeah, Louisiana was, I know, it was statistically one of the worst. It's very sad to see how things developed during that time. Although the work of Semmelweis and Snow successfully showed the role of sanitation in preventing infectious disease, the cause of disease was not fully understood. The subsequent work of Louis Pasteur, Robert Koch, and Joseph Lister would further substantiate the germ theory of disease. Man, how do you, like, I feel like they did a really good job showing, like, you wash hands, less disease. See the correlation? And like, they still weren't taken seriously. While studying the causes of beer and wine spoilage in 1856, I forget that Pasteur he just cared about alcohol. Oh, yeah. I think it was really tough for a lot of families, especially if they don't have. Sometimes they don't have room to do that kind of isolation, so then the whole family just ends up getting it. It's rough. It was a rough time. I was just living alone, so I just isolated by myself for most of that pandemic experience. Pasteur discovered properties of fermentation by microorganisms. He had demonstrated with his swan neck flask experiments that airborne microbes, not spontaneous generation, were the cause of food spoilage, and he suggested that if microbes were responsible for food spoilage and fermentation, they could also be responsible for causing infection. This guy was on a mission. He was here to step on the necks of all these scientists who were like naysaying a bunch of theories. He said, no, germs don't just spawn out of nowhere. And now he's also going to say, germs exist and they cause infection. And you were in high school in it? Yeah. Oh, it feels like I know for like a lot of young people, it was a really rough time. I was in a grad school, so it wasn't as like not much of a developmental issue for me. But it did get in the way of doing lab research for a bit. Because you can't really get in there. You can't go to the lab during the pandemic unless you like coordinate. You mostly played video games. Hey, that's good. I'm happy when people said they just got to do their hobbies. British surgeon Joseph Lister was trying to determine the causes of post-surgical infections. Many physicians did not give credence to the idea that microbes on their hand, on their clothes, or in the air could infect patients' surgical wounds, despite the fact that 50% of surgical patients on average were dying of post-surgical infections. Oh my gosh. If you were getting a surgery back then, you just had like a 50-50 odd of just dying. Man. Lister, however, was familiar with the work of Semmelweis and Pasteur. Therefore, he insisted on hand washing and extreme cleanliness during surgery. In 1867, to further decrease the incidence of post-surgical wound infections, Lister began using carbolic acid, phenol, spray disinfectant, antiseptic during surgery. His extremely successful efforts to reduce post-surgical infection caused his techniques to become a standard in medical practice. Wow. I feel like I'm sure when he introduced how cleanly he's trying to be to other physicians, they were like, you're doing way too much work. Because it sounds like back then they would just put people on the surgery bed and say, all right, I'm just cutting you open, and then they move on to the next person without washing their hands, changing their clothes, changing their instruments. They were just cutting people up one by one, just going blip 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 blip. No wonder they were just dying. Yeah, and the war amputations were definitely similar. But you would think, like, well, there's no war going on. They have all the time to, like, they're just in a hospital. They should, like, clean up, maybe. You would think they were not trying to, like, go super fast. It's not like there's wartime, like, it's not a pressing matter. But it sounds like they just didn't care. They were just cutting people open with the same things. Not even washing their hands. That part's insane to me. They cut someone open and wouldn't wash their hands. They would cut people open in autopsy and then go touch pregnant women. Insane. That's in the 1867. A few years later, Robert Koch proposed a series of postulates, Koch's postulates. This is like, again, we're back in biology 101. Chainsaw was originally invented for C-sections. Yeah, I know that one. People are stupid then. Yeah, I mean, they're stupid. I would say maybe they're more ignorant. It's not, to me, like, and I don't even mean it in a malicious way. They were just being incredibly ignorant. And I also understand, can you imagine how hard it is to, like, spread information in 1867? It's not like nowadays. You can't just Google. There's no, like, PubMed where you can search up the latest research being done. You just have to spread your information through word of mouth and hope people believe what you said. It just sucks they didn't take it seriously. I feel like if I was a doctor then, I would think, maybe I should do something. My patients keep dying. Oh, I don't know about ignorances. I feel like there's information that's I don't like knowing, but knowledge is power. I'd rather know something than not know it. Using these postulates, Koch and his colleagues were able to definitively identify that causative pathogens of specific diseases, including anthrax, tuberculosis, cholera. Koch's one microbe, one disease concept was a culmination of the 19th century's paradigm shift away from the miasma theory and towards the germ theory of disease. Koch's postulates are discussed more thoroughly in how pathogens cause disease. One microbe, one disease. Interesting. Oh, clinical focus part 2. After suffering a fever, congestion, cough, and increasing aches and pains for several days, Barbara suspects that she has the case of the flu. She decides to visit the health center at her university. The PA tells Barbara that her symptoms could be due to a range of diseases such as influenza, bronchitis, pneumonia, or tuberculosis. During her physical examination, the PA notes that Barbara's heart rate is slightly elevated. Using a pulse oximeter, a small device that clips on her finger, he finds that Barbara has hypoxemia, a lower than normal level of oxygen in the blood. Using a stethoscope, the PA listens for abnormal sounds made by Barbara's heart, lung, and digestive system. And as Barbara breathes, the PA hears a crackling sound and notes a slight shortness of breath. He collects a sputum sample, noting the greenish color of the mucus, and orders a chest radiograph, which shows a shadow in the left lung. All of these signs are suggestion of pneumonia, a condition in which the lung fills with mucus. Poor Barbara. She thought she was coming in here just to get her flu checked out, and she's got pneumonia. I hope she doesn't have a sad case. These, like, clinical focus things always tend to have... I feel like it's been 50-50 if it's sad or not. This is why it's important to go to the doctor. Go to the doctor if you feel like you've got something wrong with you. You don't want to just sit around and think you have a flu when you actually have pneumonia. But also then, in America at least, it's not that easy to just go to the doctor. What's this? Where's the title to this figure? I guess it's just a timeline. Yeah, it does suck. It does suck to think you think you have the flu and it's just pneumonia, and it ends up being pneumonia. Unique characteristics of prokaryotic cells. Okay, so now we're off of the theoretical and we're going into, like, actually what the cells are made of. And now we're looking at prokaryotic cells. By the end of this section, we will be able to identify or be able to explain the distinguishing characteristics of prokaryotic cells, describe common cell morphologies and cellular arrangements typical of prokaryotic cells, and explain how cells maintain their morphology, describe internal and external structures of prokaryotic cells in terms of their physical structure, chemical structure, and function. Compare the distinguishing characteristics of bacterial and archaeal cells. Oh boy, cellular morphology. Cell theory states that the cell is the fundamental unit of life. However, cells vary significantly in size, shape, structure, and function. At the simplest level of construction, all cells possess a few fundamental components. These include cytoplasm, a gel-like substance composed of water and dissolved chemicals needed for growth, which is contained within a plasma membrane, also called a cell membrane or cytoplasmic membrane, one or more chromosomes, which contain the genetic blueprints of the cell, and ribosomes, organelles used for the production of proteins. It's like that is biology 101. Oh yeah, stuff like that. I feel like unfortunately so many people end up they don't like have the resources to go to a doctor or any kind of clinic to get it checked on, and they just have to stay at home hoping it gets better. And in most cases, it probably does just get better, and they'd have no idea what they might have had. But there's also a lot of scenarios where people think it's gonna get better if they stay home, and it doesn't, and it gets worse, and they might get severely sick, and there might be some fatal outcomes because of that. It's really sad. Beyond these basic components, cells can vary greatly between organisms and even within the same multicellular organism. The two largest categories of cells, prokaryotic cells and eukaryotic cells, are defined by the major difference in several cell structures. Prokaryotic cells lack a nucleus surrounded by a complex nuclear membrane and generally have a single circular chromosome located in a nucleoid. Nucleoid. Eukaryotic cells have a nucleus surrounded by a complex nuclear membrane that contains multiple rod-shaped chromosomes. All plant cells and animal cells are eukaryotic. Some microorganisms are composed of prokaryotic cells, whereas others are composed of eukaryotic cells. Prokaryotic microorganisms are classified within the domains archaea and bacteria, whereas eukaryotic organisms are classified within the domain eukarya. Eukarya. Oh man, chapter 1 was all about classification, and there was I feel like I just went over that last week, and my brain is now blank again. It's blank on what all the different domains and genuses were again. Yeah, I like the word nucleoid. Nucleoid. The structures inside a cell are analogous to the organs inside a human body, with unique structures suited to specific functions. Some of the structures found in prokaryotic cells are similar to those found in some eukaryotic cells. Others are unique to prokaryotes. Although there are some exceptions, eukaryotic cells tend to be larger than prokaryotic cells. The comparatively larger size of eukaryotic cells dictates the need to compartmentalize various chemical processes within different areas of the cell. Using complex membrane-bound organelles, in contrast, prokaryotic cells generally lack membrane-bound organelles. However, they often contain inclusions that compartmentalize their cytoplasms. Figure 3.12 illustrates structures typically associated with the prokaryotic cells. These structures are described in more detail in the next section. Ooh, look. It's like, where is it? There's the flagella, ileus, fimbriae, plasma cell membrane, cell wall, capsule. It's kind of, I really like this picture. The nucleoid, the inclusion, the plasmids, the cytoplasm. It's a good diagram. I distinctly remember making something similar in elementary school as well. Common cell morphologies and arrangements. Individual cells of a particular prokaryotic organisms are typically similar in shape or cell morphology. Although thousands of prokaryotic organisms have been identified, only a handful of cell morphologies are commonly seen microscopically. Figure 3.13 names and illustrates cell morphologies commonly found in prokaryotic cells. In addition to cellular-shaped prokaryotic cells of the same species may group together in certain distinctive arrangements, depending on the plane of cell division. Some common arrangements are shown in figure 3.14. It's kind of like we kind of went over this briefly in chapter 1. The coccus, the cocci. Round. I made a cell itself out of a large styrofoam ball that I got at a Hobby Lobby. Oh, that's a cool like model. I feel like I don't think I did anything anywhere close to that creative in elementary school. I remember just drawing. We drew one, and it definitely did not look good. The bacilli, the rod, the vibrio, the curved rod, curved rod, curved rod, and then the coccobacilli. They took the round, they took the rod, and they make it a short rod. Shortened rod. Spirilla, spiral. A curly stuff, I'm really bad at pronouncing this one. Spirochetti. Long, loose helical spiral. Aren't the fun? Like, I love bacteria shapes. I think they're so cool. I'm a big fan of the spirally, curly ones. Oh, and then there are arrangements of it. The coccus, the single coccus, the pair of cocci, grouping of 4 cells, the tetrad, the streptococcus, the staphylococcus, the bacillus, the diplobacillus, the streptobacillus, the palisade. Kind of crazy. There's so many different arrangements. These are only the common ones. There's way more, but these are the most common things you'll see. Yeah, it does look like a noodle. It's very noodly. In most prokaryotic cells, morphology is maintained by the cell wall in combination with cytoskeletal elements. The cell wall is a structure found in most prokaryotes and some eukaryotes. It envelops the cell membrane, protecting the cell from changes in osmotic pressure. Osmotic pressure occurs because of differences in the concentration of solutes on opposing sides of a semi-permeable membrane. Water is able to pass through a semi-permeable membrane, but solutes, dissolved molecules like salts, sugars, and other compounds, cannot. When the concentration of solutes is greater on one side of the membrane, water diffuses across the membrane from the side with the lower concentration, more water to the side with a higher concentration, less water, until the concentration of both sides become equal. This diffusion of water is called osmosis, and it can cause extreme osmotic pressure on a cell wall when its external environment changes. Oh no, not the strep throat. Yeah, strep throat is so nasty. I actually have never gotten it, but it's not very pleasant. I know that. I've seen enough of it to know that it's not very pleasant. The external, I wouldn't recommend it. Yeah. I hope I can continue to avoid the strep throat. The external environment of a cell can be described as an isotonic, hypertonic, or hypotonic medium. In an isotonic medium, it's like we're getting into like general chemistry, the most baby general chemistry. In an isotonic medium, the solute concentrations inside and outside the cell are approximately equal, so there is no net movement of water across the cell membrane. In an hypertonic medium, the solute concentration outside the cell exceeds that inside the cell, so water diffuses out of the cell and into the external medium. In a hypotonic medium, the solute concentration inside the cell exceeds that outside of the cell, so water will move by osmosis into the cell, and this causes the cell to swell, potentially lyse or burst. Yeah, I know like tonsil removal, that's like, yeah, it's one of the big reasons why people, they get rid of their tonsils because they don't want the strep throat anymore. The degree to which a particular cell is able to withstand changes in osmotic pressure is called tonicity. Cells that have a cell wall that are better able to withstand subtle changes in osmotic pressure and maintain their shape. In hypertonic environments, cells that lack a cell wall can become dehydrated, causing crenation or shriveling of the cell. Shrivel. The plasma membranes contract and appear scalloped or notched. By contrast, cells that possess a cell wall undergo plasmolysis rather than crenation. In plasmolysis, the plasma membrane contracts and detaches from the cell wall, and there's a decrease in interior volume, but the cell wall remains intact, thus allowing the cell to maintain some shape and integrity for a period of time. Likewise, cells that lack a cell wall are more prone to lysis in hypotonic environments. The presence of a cell wall allows the cell to maintain its shape and integrity for a longer time before lysing. I'll be honest, I am sure I was taught this in biology class, but I didn't know the terms crenation and plasmolysis. It's probably deep in the recesses of my mind. I just don't remember those terms specifically. Funny round shape into funny not round shape. Thing go in and out. Water go in and out. Solute out, water in. It's kind of funny to see. The nucleoid. All cellular life has a DNA genome. Oh, you haven't taken a biology class yet? You will learn many things then. I'm sure whenever you end up taking like a college biology class, you'll hear a lot of these terms because this is like biology 101, especially these sections. The later chapters are probably more deep into like microbiology, which you may or may not get into, but these are like really general bio topics. The nucleoid. All cellular life has a DNA genome organized into one or more chromosomes. Prokaryotic chromosomes are typically circular, haploid, unpaired, and not bound by a complex nuclear membrane. Prokaryotic DNA and DNA-associated proteins are concentrated within the nucleoid region of the cell. In general, prokaryotic DNA interacts with nucleoid-associated proteins, NAPs, that assist in the organization and packaging of the chromosome. In bacteria, NAPs function similar to histones, which are the DNA organizing proteins found in eukaryotic cells. In archaea, the nucleoid organization is either by NAPs or histone-like DNA organizing proteins. Oh, if you don't have many biology courses, and you might not see a lot of this stuff. If you would only, maybe if you take only just Bio 101, but I know you said you're studying geology, so maybe you don't take Bio 101. The nuclear region, the area enclosed by the green dashed line, is a condensed area of DNA found within the prokaryotic cells. Because of the density of the area, it does not readily stain and appears lighter in color when viewed with a transmission electron microscope. It is kind of funky. Plasmids. Prokaryotic cells may also contain extra chromosomal DNA or DNA that is not part of the chromosome. This extra chromosomal DNA is found in plasmids, which are small, circular, double-stranded DNA molecules. Cells that have plasmids often have hundreds of them within a single cell. Plasmids are most commonly found in bacteria. However, plasmids have been found in archaea and eukaryotic organisms. Plasmids often carry genes that confer advantageous traits such as antibiotic resistance. Thus, they are important to the survival of the organism. We will discuss plasmids in more detail in mechanisms of microbial genetics. Oh boy, plasmids. Whenever we get to that section, you already know it's going to get into so much depth. Plasmids are they're basically the basis of most biological experiments. You kind of take advantage of the plasmids and introduce what you use plasmids that contain the information you want that bacteria to have, and then you insert that into the bacteria and say, you need this information, you need this level of antibiotic resistance, or I need you to make this specific protein. It's really interesting how we can hijack their machinery, basically. Ribosomes. All cellular life synthesizes proteins and organisms in all three domains of life possess ribosomes, structures responsible for protein synthesis. However, ribosomes in each of the three domains are structurally different. Ribosomes themselves are constructed from proteins along with ribosomal RNA. RNA. Prokaryotic ribosomes are found in the cytoplasm. They are called the 70S ribosomes because they have the size of They have a size of 70s. Oh, makes sense. Whereas eukaryotic cytoplasmic ribosomes have a size of 80s. S stands for Svedberg unit, a measure of sedimentation in an ultracentrifuge which is based on size, shape, and surface qualities of the structure being analyzed. I didn't know that's what S stood for. Interesting. Although they are the same size, bacterial and archaeal ribosomes have different proteins and rRNA molecules, and the archaeal versions are more similar to their eukaryotic counterparts than those found in bacteria. I've checked. Oh, you only need 1 4-credit biology course. Yeah. You might like take like a very basic, maybe a basic biology class. Maybe they would. I don't know if they would let you take a more advanced one if you don't have a basic one done. Usually, at least in my university experience, it's like you wouldn't have the prerequisites. No, not sedimentary rocks. Sedimentation. Sedimentation. That word is pretty common actually in biology because you use a centrifuge, which is a machine that spin very very fast, and things start to they deposit into the bottom of like your tube or something. They sediment at the bottom. Inclusions. As single-celled organisms living in unstable environments, some prokaryotic cells have the ability to store excess nutrients within cytoplasmic structures called inclusions. I don't know much about prokaryotic. Yeah. I mean, it's not a completely unique. Yeah, it's not a completely unique term. They definitely just mean the same thing, but applied in the different field. No rocks in biology. Well, there's rocks, but no rocks here. Storing nutrients in a polymerized form is advantageous because it reduces the buildup of osmotic pressure that occurs as all cell accumulates solutes. Various types of inclusions store glycogen and starches, which contain carbon that cells can access for energy. Volutin granules, also called metachromatic granules. That's so much easier to pronounce than volutin. Because of their staining characteristics, are inclusions that store polymerized inorganic phosphate that can be used in metabolism and assist in the formation of biofilms. Microbes known to contain metachromatic granules include the archaea Methanosarcina, the bacterium Coronae, bacterium diphtheriae, and the unicellular eukaryotic alga, Chlamydomonas. Okay. I am not familiar with any of these microbes. I'm sorry, microbes. I'm butchering your names. Sulfur granules, another type of inclusion, are found in sulfur bacteria of the genus Theobacillus. These granules store elemental sulfur, which the bacteria use for metabolism. Occasionally, certain types of inclusions are surrounded by a phospholipid monolayer. Embedded with protein. Latin is hard. It's just so impossible to pronounce. And sometimes you see what they mean, and then and sometimes I'm just I really don't know what you're trying to say. I get Theobacillus. Theo is that's sulfur. Oh, you're really good at it. Well, you would succeed. You would succeed in biology. There's so much of it. Though sometimes it feels like I don't even know where these names are coming from. Some of them are more obvious than others. But I am no expert in nomenclature. A lot of at least the super well known bacteria I'm much more familiar. Yeah, paleontology. It does make sense. Polyhydroxybutyrate, PHB, which can be produced by the species of Bacillus and Pseudomonas, is an example of inclusion that displays this type of monolayer structure. Industrially, PHB has also been used as a source of biodegradable polymers for bioplastics. Interesting. PHB. PHB. Bioplastics. Biodegradable polymer for bioplastics. Like, I wonder how effective it is. I have a feeling it's not it is not as biodegradable as I would hope. It's a good explanation what paleontology is, because I feel like sometimes a lot of people might have misconceptions of what paleontology really includes. I think most people just think dinosaurs. And I like just specifically dinosaur. Just only dinosaur. Maybe even like they just think of T-Rex. Some prokaryotic cells have other types of inclusions that serve purposes other than nutrient storage. For example, some prokaryotic cells produce gas vacuoles, accumulation of small protein-lined vesicles of gas. These gas vacuoles allow the prokaryotic cells that synthesize them to alter their buoyancy so that they can adjust their location in the water column. Magnetotactic bacteria such as Magnetospirillum magnetotacticum contain magnetosomes. I'm saying magnet a lot, which are inclusions of magnetic iron oxide or iron sulfide surrounded by a lipid layer. These allow cells to align along a magnetic field, aiding their movement. Cyanobacteria such as Anabaenia cylindrica and bacteria such as Halotheobacillus neapolitanus produce carboxome inclusions. Carboxysomes are composed of outer shells of thousands of protein subunits. Their interior is filled with ribulose 1,5-biphosphate carboxylase oxygenase, Rubisco. Rubisco, I know that. And carbonic anhydrase. Both of these components are used for carbon metabolism. Some prokaryotic cells also possess carboxyles that sequester functionally related enzymes in one location. These structures are considered proto-organelles because they compartmentalize important compounds or chemical reactions, much like many eukaryotic organelles. Yeah, there's way more than just dinosaurs. It's a bit unfortunate that people only think about, like, when I even say dinosaurs, I feel like it's maybe too. You need to simplify maybe even further of like the most simple dinosaur there is. Endospores. Bacterial cells are generally observed as vegetative cells, but some genera of bacteria have the ability to form endospores, structures that essentially protect the bacterial genome in a dormant state when environmental conditions are unfavorable. Endospores, not to be confused with the reproductive spores formed by fungi. I feel like there could have been a better word for endospores, because I tend to think of the reproductive spores of fungi. I feel like that's a much more common word, like, that's where my brain goes for endospores. Allows some bacterial cells to survive long periods without food or water, as well as exposure to chemicals, extreme temperatures, and even radiation. Table 3.1 compares the characteristics of vegetative cells and endospores. Vegetative cells, endospores. They are sensitive to extreme temperatures and radiation, resistant, gram-positive, do not absorb gram stain, dehydrated. Interesting. The process by which vegetative cells transform into endospores is called sporulation, and this generally begins when nutrients become depleted or environmental conditions become otherwise unfavorable. That's true. I need to hydrate. Thank you. I will hydrate. The process begins with the formation of a septum in the vegetative bacterial cell. The septum divides the cell asymmetrically, separating a DNA 4 spore from the mother cell. Another hydration. Okay, I'll take another sip. The 4 spore, which will form the core of the endospore, is essentially a copy of the cell's chromosomes and is separated from the mother cell by a second membrane. I feel like I don't remember any of this. Lots of learning and yapping, extra hydration, not bad. That's true. I have a giant jug of water. I did get myself like a huge cup of water to prepare for the yapping and reading. A protein coat then forms around the cortex while the DNA of the mother cell disintegrates. Further maturation of the endospore occurs with the formation of an outermost exosporium. The endosphere is released upon disintegration of the mother cell, completing sporulation. I don't remember any of this. That's crazy. I feel like in my classes they did not care about endospores. Oh wow. Sporulation begins following asymmetric cell division. The forespore becomes surrounded by a double layer of membrane, a cortex, and a protein coat before being released as a mature endospore upon disintegration of the mother cell. I like the term mother cell is very funny. An electron micrograph of carboxydothermous hydrogenoformans endospore. These Bacillus spirilla cells are undergoing sporulation. The endospores have been visualized using malachite green cells. Ah, look at, you can see the little green, you can see the endospores. Fascinating. Fascinating work. Endospores of certain species have been shown to persist in a dormant state for extended periods of time, up to thousands of years. What the frick? What the frick? Thousands? However, when living conditions improve, endospores undergo germination, re-entering a vegetative state. After germination, the cells become metabolically active again and is able to carry out all its normal functions, including growth and cell division. Wow. You mean can I do that? Can I undergo endospore? Like, I wanna go dormant. Not all bacteria have the ability to form endospores. However, there are a number of clinically significant endospore-forming gram-positive bacteria of the genera Bacillus and Clostridium. These include B. anthracis, the causative agent of anthrax, which produce endospores capable of surviving for many decades. C. tetani, which causes tetanus. C. difficile, and C. perfringens causes gangrene, and C. botulinum. Oh, botulism. Oh yeah, I have heard of plants. Yeah, I've heard of plants doing that. That's true. I guess it's not like super unheard of. I feel like I'm just surprised that I don't recall learning about this bacteria. But then also when I'm reading this, I understand this. Surely, like, I knew that this happens because all these diseases that they're talking about, that's one of the big problems, is they just survive. They just live for many decades, like botulism. Pathogens such as these are particularly difficult to combat because their endospores are so hard to kill. Special sterilization methods for endospore-forming bacteria are discussed in control of microbial growth. Yeah. Yeah, that was like a big problem with anthrax, especially. There's not much you can do about anthrax. Plasma membrane. Structures that enclose the cytoplasm and internal structures of the cell are known collectively as the cell envelope. In prokaryotic cells, the structure of the cell envelope varies depending on the type of cell and organism. Most, but not all, prokaryotic cells have a cell wall. The makeup of the cell wall varies. All cells, prokaryotic, eukaryotic, have a plasma membrane, also called cytoplasmic membrane or cell membrane, that exhibits selective permeability, allowing some molecules to enter or leave the cell while restricting the passage of others. Very fluid. Woo! Go in, go out, go in, go out. The structure of plasma membrane is often described as the fluid mosaic model. Oh, yeah, exactly. That's what I was saying. Very fluid, which refers to the ability of membrane components to move fluidly within the plane of the membrane, as well as the mosaic-like composition of the components, which include a diverse array of lipid and protein components. The plasma membrane structure of most bacterial and eukaryotic cell types is bilayer, composed mainly of phospholipids formed with ester linkages and proteins. These phospholipids and proteins have the ability to move laterally within the plane of the membrane as well as between the two phospholipid layers. So these things can be transported in, out, but they can also move side to side. a lot of if you're doing a lot of times with like bacterial research and like lab experiments, you're usually looking for things that are found in this layer. Sometimes you want to like extract a receptor or protein found here, and so then you have to think of ways like how do we get how do we get it? How are we going to get it? That was my undergrad research thesis. Well, not thesis. It was a project. There was a protein that was on the this biphosphate this layer, and we wanted to extract it. And it was actually really hard. I don't think I ever successfully did it. Especially the difficulty comes when it's both in and out, and that's what we were looking at. Archaeal membranes are fundamentally different from bacterial and eukaryotic membranes in a few significant ways. First, archaeal membrane phospholipids are formed with ester linkages in contrast to the ester linkages found in bacterial and or eukaryotic cell membranes. Second, archaeal phospholipids have branched chains, whereas those of bacterial and eukaryotic cells are straight chain. Finally, some archaeal membranes can be formed of bilayers like those found in bacteria and eukaryotes. Other archaeal plasma membranes are lipid monolayers. Well, we have an ad break coming up. I think I will use this time to do a little stretchy stretch because I've been sitting for like 3 hours and I need to stretch my legs and also maybe get a little bit more water. So I will be right back. I'm gonna go to the BRB. BRB. Hi. I walked around my apartment for like 3 minutes. Stretching the legs. Stretch leg good. Stretch leg good. Don't get blood clots. Blood clot bad. Proteins on the... Where are we? I think here. Proteins on the cell's surface are important for a variety of functions, including cell-to-cell communication and sensing environmental conditions and pathogenic virulence factors. Membrane proteins and phospholipids may have carbohydrates, sugars, associated with them and are called glycoproteins or glycolipids, respectively. These glycoprotein and glycolipid complexes extend out from the surface of the cell, allowing the cell to interact with the external environment. Glycoproteins and glycolipids in the plasma membrane can vary considerably in chemical composition among archaea, bacteria, and eukaryotes, allowing scientists to use them to characterize unique species. Yeah, it's a huge part of understanding why certain species are the way what they are because of just what's expressed on their phospholipid layer. It's a huge tell. There's so many different proteins. There are so many different receptors. Holy. Plasma membranes from different cell types can also contain unique phospholipids, which contain fatty acids. As described in using biochemistry to identify microorganisms, phospholipid-derived fatty acid analysis PLFA profiles can be used to identify unique types of cells based on differences in fatty acids. Archaea, bacteria, and eukaryotes each have a unique PFLA profile. Oh boy, membrane transport mechanisms. One of the most important functions of the plasma membrane is to control the transport of molecules into and out of the cell. Internal conditions must be maintained within a certain range despite any changes in the external environment. The transport of substances across the plasma membrane allows cells to do so. It's actually, I always thought like the transport mechanisms are pretty fascinating. And they're actually pretty complicated. You think it's just like go in and out, but it's so tightly regulated. It's so orchestrated, kind of orchestrated beautifully. You have to give it to the cells. They've created something wonderful. Cells use various modes of transport across the plasma membrane. For example, molecules moving from a higher concentration to a lower concentration when the concentration gradient are transported by simple diffusion, also known as passive transport. I would like to be passively transported. Some small molecules, like carbon dioxide, may cross the membrane bilayer directly by simple diffusion. However, charged molecules as well as large molecules need the help of carriers or channels in the membrane. These structures ferry molecules across the membrane, a process known as facilitated diffusion. Active transport occurs when cells move molecules across their membrane against the concentration gradient. A major difference between passive and active transport is that active transport requires adenosine triphosphate, ATP. Give it up for our boy ATP. The key building block of all of us. Or other forms of energy to move molecules uphill. Therefore, active transport structures are often called pumps. They're pumping the things out. Or in. Facilitated diffusion. Take thing, help thing, receptor. Yep. These receptors, these protein channels, there's usually a lot of interest in studying them. Active transport against a concentration gradient. You haven't heard about ATP since high school biology? I mean, yeah. I don't think it's not something that really comes up in conversation. Extracellular fluid. It's the sodium-potassium channel pumps. Sodium-potassium channel pumps. It's crazy how many things are controlled by these kind of controlled by this interaction, this concentration gradient. Group translocation also transports substances into bacterial cells. In this case, as a molecule moves into a cell against its concentration gradient, it is chemically modified so that it does not require transport against an unfavorable concentration gradient. A common example of this is the bacterial phosphate Bacterial phosphotransferase system, a series of carriers that phosphorylates, i.e. adds phosphate ions to, glucose or other sugars upon entry into cells. Since the phosphorylation of sugars is required during the early stages of sugar metabolism, the phosphotransferase system is considered to be an energy neutral system. No energy used. You joined the Discord. Oh, thank you. An official bacteria bud. Don't worry. You don't need to join the Discord to be an official bacteria bud. You know, just being here, lurking, you don't even have to talk. If you're just here lurking, I consider you. I consider you a bacteria bud. You have no choice. I appreciate you. I appreciate you yapping. I appreciate anyone that yaps, doesn't yap, just hangs out, just lurks. All of you are appreciated. I see the silent viewers and know you are appreciated. Photosynthetic membrane structures. Some prokaryotic cells, namely cyanobacteria and photosynthetic bacteria, have membrane structures that enable them to perform photosynthesis. Oh, thank you for the follow, Liminal. These structures consist of an infolding of the plasma membrane that encloses photosynthetic pigments such as green chlorophylls and bacterial chlorophylls in cyanobacteria. These membrane structures are called thylakoids. Thylakoids in photosynthetic bacteria, and they are called chromatophores, lamellae, or chlorosomes. Hello, Liminal. You're a yapper both online and IRL. I am too, which is why I started streaming anyway. Streaming is just a way to yap, have an excuse to yap, I should say. If you forgot, oh, thank you for checking in, Grin. That is a really cute emote. Let's see. Now we're looking at the cell wall. The primary function of the cell wall is to protect the cell from the harsh conditions in the outside environment. It keeps us safe. Wall keeps safe. When present, there are notable similarities and differences among the cell walls of archaea, bacteria, and eukaryotes. The major component of bacterial cell walls is called peptidoglycan, or murin. Who's calling that? Who's calling peptidoglycan that? It is only found in bacteria. Structurally, peptidoglycan resembles a layer of meshwork of fabric. Each layer is composed of long chains of alternating molecules of N-acetylglucosamine. That's my tongue cannot pronounce it. Yeah, wall keep bad stuff out. Wall good. And N-acetylmuramic acid. NAM. NAG and NAM. The structure of the long chains has significant two-dimensional tensile strength due to the formation of peptide bridges that connect NAG and NAM within each peptidoglycan layer. In gram-negative bacteria, tetrapeptide chains extending from each NAM unit are directly cross-linked, whereas in gram-positive bacteria, these tetrapeptide chains are linked by pentaglycine cross bridges. Peptidoglycan subunits are made inside of the bacterial cell and then exported and assembled in layers, giving the cell its shape. Very cool. It's impressive how sophisticated the construction of these cells are. So tiny, yet so intricate. So beautifully put together. Since peptidoglycan is unique to bacteria, many antibiotic drugs are designed to interfere with the peptidoglycan synthesis, weakening the cell wall and making bacterial cells more susceptible to the effects of osmotic pressure. They're killing the wall. The drugs are killing the wall, which makes sense. Makes sense how the drugs work. In addition, certain cells of the human immune system are able to recognize bacterial pathogens by detecting peptidoglycan on the surface of a bacterial cell. These cells then engulf and destroy the bacterial cell using enzymes such as lysozymes, which breaks down and digests the peptidoglycan in their cell walls. Nag, nam. Nag, nam. Nag, nam. What a wonderful construction. It's almost, I can understand how people believe in like sophisticated design, so to speak, as in like it's kind of hard to imagine how these things happen to be in such a like it all makes sense. Like we we have mechanisms to handle these unique what is it molecules. Like we're just able to detect this so easily, and it's hard not to think like this is all meant to be. It's all sophisticated, but it's just really evolution. Evolution did a fantastic job getting our immune system to this point. A Gram staining protocol is used to differentiate two common types of cell wall structures. Gram-positive cells have a cell wall consisting of many layers of peptidoglycan totaling 30 to 100 nanometers in thickness. I too love my white blood cells. I appreciate what I appreciate what my immune system is going through every single day. Because I don't know who knows what battles they're fighting, even right now. These peptidoglycan layers are commonly embedded with teichoic acids, TAs, carbohydrate chains that extend through and beyond the peptidoglycan layer. TA is thought to stabilize peptidoglycan by increasing its rigidity. I distinctly remember this being a question in my bio exam. I don't know why. The deep inside my brain, one of my cells just remembered that this was a question. TA also plays a role in the ability of pathogenic gram-positive bacteria such as Streptococcus to bind to certain proteins on the surface of host cells, enhancing their ability to cause infection. I will hydrate. Hold on. I know Streptococcus keeps coming back. It's hard to avoid it. Streptococcus is like it's a big dog in bacteria. It's a big dog. It's like E. coli. It's a celebrity, so to speak. In addition to peptidoglycan TAs, bacteria of the family Mycobacteriaceae have an external layer of waxy mycolic acids in their cell wall, as described in staining microscopic specimens. These bacteria are referred to as acid-fast, since acid-fast stains must be used to penetrate the mycolic acid layer for purposes of microscopy. Yeah. Yeah, I think anyone who ever had strep throat probably has beef with Streptococcus. I think he's got many ops. Bacteria contain two common cell wall structures. Gram-positive cell walls are structurally simple, containing a thick layer of peptidoglycan with embedded TA external to the plasma membrane. Gram-negative cell walls are structurally more complex, containing a thin layer of peptidoglycan and an outer layer membrane containing lipopolysaccharide. Wow, it's kind of crazy how much more complicated that is. This gram-positive cell that they're just kind of chilling with just TA, and then there is a bunch of things happening right here with the gram-negative. 2 to 3 times a year. Okay, I get it. You can hate Streptococcus. You are free to hate strep. Number one enemy. Gram-negative cells have a much thinner layer of peptidoglycan, no more than 4 nanometers thick, than gram-positive cells, and the overall structure of their cell envelope is more complex. In gram-negative cells, a gel-like matrix occupies the periplasmic space between the cell wall and the plasma membrane. And there is a second lipid bilayer, another lipid bilayer. Who would have thought that the bilayer had another membrane? Called the outer membrane, which is external to the peptidoglycan layer. This outer membrane is attached to the peptidoglycan by murine lipoprotein. The outer leaflet of the outer membrane contains the molecule lipopolysaccharide, LPS, which functions as an endotoxin in infections involving gram-negative bacteria, contributing to symptoms such as fever, hemorrhaging, and septic shock. I'm just shouting out LPS because I did a lot of study on this as an undergrad student. We were doing a lot of research on like sepsis, which is when the bacterial infection gets into your blood. it's usually really, really bad. Really, really bad. And it's caused by this little guy. Each LPS molecule is composed of lipid A, a core polysaccharide, and an O side chain that is composed of sugar-like molecules that comprise the external face of the LPS. The composition of the O side chain varies between different species and strains of bacteria. Parts of the O side chain, called antigens, can be detected using serological or immunological tests to identify specific pathogenic strains like Escherichia coli O157H7, a deadly strain of bacteria that causes bloody diarrhea and kidney failure. Pretty nasty. I worked specifically with this strain of bacteria, and we had to be really careful, or else you could get really sick. I think most of the strains that we worked with, I only worked with it briefly because it's pretty dangerous. It's not like it's gonna kill you. You likely won't die from it, but you could get sick, and being sick from science is never good. Ooh, O antigen, the core, the lipid A, and the fatty acid. Archaeal cell wall structures differ from that of bacterial in several significant ways. First, archaeal cell walls do not contain peptidoglycan. Instead, they contain a similar polymer called pseudopeptidoglycan, pseudomurin, in which NAM is replaced with a different subunit. Other archaea may have a layer of glycoproteins or polysaccharides that serve as a cell wall instead of pseudopeptidoglycan. Last, as is the case with some bacterial species, there are a few archaea that appear to lack cell walls entirely. No cell wall. No cell wall. Nothing to protect them. Glycosylases and S layers. Although most prokaryotic cells have cell walls, some may have additional cell envelope structures exterior to the cell wall, such as glycosyl. My brain can't comprehend how to pronounce that, even though I know exactly how to pronounce that. In my head, I'm hearing it, but my brain to my mouth is not. It's not cooperating. And S layers. A glycocalyx is a sugar coat of which there are two important types: capsules and slime layers. The slime layer. A capsule is an organized layer located outside of the cell wall and usually composed of polysaccharides or proteins. A slime layer is a less tightly organized layer that is only loosely attached to the cell walls and can be more easily washed off. Slime layers may be composed of polysaccharides, glycoproteins, or glycolipids. The slimy microbes are pretty slimy. That's usually it's a good tell of like if something's gone bad, or especially meat. If meat's gone bad, it's probably slimy. They just love being slimy, guys. Slimy boys. Glycocalyces allow cells to adhere to surfaces, aiding in the formation of biofilms. Oh, see, this is exactly what we were talking about, biofilms. Colonies of microbes that form in layers on surfaces. In nature, most microbes live in mixed communities within biofilms, partly because the biofilm affords them some level of protection. Biofilms generally hold water like a sponge, preventing desiccation, aka they won't become dehydrated. They also protect cells from predation and hinder the action of antibiotics and disinfectants. Biofilms are such a huge problem. Who would have thought bacteria together strong? All of these properties are advantageous to the microbes living in a biofilm, but they present challenges in a clinical setting where the goal is often to eliminate microbes. They want to get rid of me, but they can't because I'm too strong. Hey, these are the same figures from earlier. The ability to produce a capsule can contribute to a microbe's pathogenicity because the capsule can make it more difficult for phagocytotic cells, such as white blood cells, to engulf and kill the microorganism. Streptococcus pneumoniae, for example, produces a capsule that is well known to aid in this bacterium's pathogenicity. As explained in staining microscopic specimens, capsules are difficult to stain from microscopy. Negative staining techniques are typically used. No! We can't keep... Stop throwing me into the autoclave. Stop. No autoclave. No more. An S layer is another type of cell envelope structure. It is composed of a mixture of structural proteins and glycoproteins. In bacteria, S layers are found outside the cell wall, but in some archaea, the cell S layer serves as the cell wall. The exact function of S layers is not well It's not entirely understood, and they are difficult to study, but available evidence suggests that they may play a variety of functions in different prokaryotic cells, such as helping the cell withstand osmotic pressure and for certain pathogens interacting with the host immune system. Oh, part 3. What's going to happen to Barbara? She just got pneumonia. Or she got diagnosed with pneumonia. Is she going to get better? We're going to find out. After diagnosing Barbara with pneumonia, the PA writes her a prescription for amoxicillin, a commonly prescribed type of penicillin derivative. Go amoxicillin. I feel like whenever I get prescribed antibiotics, that's definitely the one I get. More than a week later, despite taking the full course as directed, she took the full course. Remember, guys, you have to take all your antibiotics. You can't just stop taking the antibiotics just because you think you're done. You gotta take the whole bottle. Barbara still feels weak and is not fully recovered. Although she is able to get through her daily activities, she returns to the health center for a follow-up visit. Many types of bacteria, fungi, and viruses can cause pneumonia. Amoxicillin targets the peptidoglycan of bacterial cell walls. Since amoxicillin has not resolved Barbara's symptom, the PA concludes that the causative agent probably lacks peptidoglycan, meaning that the pathogen could be a virus, a fungus, or a bacterium that lacks peptidoglycan. Another possibility is that the pathogen is a bacterium containing peptidoglycan but has developed resistance to amoxicillin. Dun dun dun! What could it be? Is Barbara gonna get the help she needs? Will they be able to diagnose what Barbara needs? Will she get better? Is she gonna die? Is she going to make it? Is she gonna be? Is her pneumonia gonna clear up? We gotta keep reading until we get to part 4. Filamentous appendages. Many bacterial cells have protein appendages embedded within their cell envelopes that extend outward, allowing interaction with the environment. These appendages can attach to other surfaces and transfer DNA or provide movement. Filamentous appendages include fimbriae, pilae, and flagella. I like the flagella the best. Fimbriae and pili. Fimbriae and pili are structurally similar, and because differentiation between the two is problematic, it's problematic. Problematic is like, it's a funny term to use for like this in science. These terms, yeah, they wiggle. They wiggle, are often used interchangeably. The term fimbriae commonly refers to short bristles like proteins projecting from the cell surface by the hundreds. Fimbriae enable a cell to attach to surfaces and to other cells. For pathogenic bacteria, adherence to host cell is important for colonization, infectivity, and virulence. Adherence to surfaces is also important to biofilm formation. The term pilus, commonly refers to longer, less numerous protein appendages that aid in attachment to surfaces. A specific type of pilus called the F. pilus or sex pilus is important in the transfer of DNA between bacterial cells, which occurs between members of the same generation when two cells physically transfer or exchange parts of their respective genomes. This is an intimate moment for the bacteria. Very, very intimate. We'll leave them be. Group A strep. Before the structure, it's strep again. Before the structure and function of the various components of the bacterial cell envelope were well understood, scientists were already using cell envelope characteristics to classify bacteria. In 1933, Rebecca Lansfield proposed a method for serotyping various beta hemolytic strains of Streptococcus species using an agglutination assay, a technique using the clumping of bacteria to detect specific cell surface antigens. In doing so, Lansfield discovered that one group of S. pyogenes found in Group A was associated with a variety of human diseases. She determined that various strains of Group A strep could be distinguished from each other based on variations in specific cell surface proteins that she named M proteins. M. Very creative name, Miss Lansfield. M. The M. The M. Today, more than 80 different strains of Group A strep have been identified based on M proteins. Various strains of Group A strep are associated with a wide variety of human infections, including streptococcal pharyngitis, strep throat, impetigo, toxic shock syndrome, scarlet fever, rheumatic fever, and necrotizing fasciitis. The M protein is an important virulence factor for group A strep, helping these strains evade the immune system. Changes in M proteins appear to alter the infectivity of a particular strain of group A strep. Okay. I I see. Group A. I don't understand why they were really called Group A though. Flagella. My favorite structure, the wiggly. Flagella are structures used by cells to move in aqueous environments. Bacteria flagella act like propellers. They are stiff spiral filaments composed of flagellin protein subunits that extend outward from the cell and spin in solution. The basal body is the motor of the flagellum and is embedded in the plasma membrane. A hook region connects the basal body to the filament. Gram-positive and gram-negative bacteria have different basal body configurations due to differences in cell wall structure. Insert wiggle gif, yeah. Just wiggle. Different types of motile bacteria exhibit different arrangements of flagella. A bacterium with a singular flagellum, typically located at one end of the cell, polar, is said to have a monotrichous flagellum. An example of a monotrichously flagellated bacterial pathogen is Vibrio cholerae, the gram-negative bacterium that causes cholera. Cells with amphitrichous flagella have a flagellum or tufts of flagella at the end. An example is Spiralium minor, the cause of spirally Asian rat bite fever, or Sodoku. I've never heard of that disease. Why did they name it that? Cells with Lophotrichous flagella have a tuft at one end of the cell. The gram-negative Bacillus pseudomonas aeruginosus, an opportunistic pathogen known for causing many infections, including swimmer's ears and burn wound victims, has Lophotrichous flagella. Yeah, I was just thinking sounds like Sudoku. Flagella that cover the entire surface of the bacterial cell are called petricose flagella. The gram-negative bacterium E. coli shows a petricose arrangement of flagella. I had no idea there were so many different flagella. I was just thinking of flagella. I didn't even know there were names for these. I'm not a very good bacteria. I'm not very good at Sudoku. But I like games similar to that. I'm just I like puzzle games and word games and whatever kind of games like that. I'm not. Sudoku has never been my cup of tea. There's the mono, amphi, lopho. I'm kind of digging the Petri guy. Look, it's like hair. But also I feel like I don't like amphi. I think my favorite is Petri, and then mono, then lopho, and then probably amphi is least favorite. I don't like this symmetrical nonsense going on. It's making me uncomfortable. I think it's because it looks like there's no way to identify which one's the end. Yeah, Sudoku takes a bit of practice. I think I enjoy when I am doing puzzles. I enjoy things that are more word based rather than math based. I really like crosswords. Crosswords are my go-to jam when I'm bored. I like solving the New York Times crossword. Directional movement depends on the configuration of the flagella. I get it's based on patterns. I just mean the numerical patterns don't work for my brain. I need language. I need it just doesn't it doesn't interest me enough to get into it. Bacteria can move in response to a variety of environmental signals, including light, phototaxis, magnetic fields, magnetotaxis, using magnetosomes, and most commonly chemical gradients, chemotaxis. I always like the different taxis. I think the word taxis as a root word is really funny. Hi, Beppo. Hi. Thank you for dropping by. Using a process of elimination. Yeah, I guess that makes sense. I'm sure if I like sat down and practiced it, like understood it better, I would get into it. But I just don't think I would ever really like. I just I'm already busy with enough, and I think crosswords usually take most of my time anyway. If I was bored to do, if I wanted to do something with my brain. Purposeful movement towards a chemical attractant like a food source or away from a repellent like a poisonous chemical is achieved by increasing the length of runs and decreasing the length of tumbles. Run and tumble. Run and tumble. That's all bacteria do. They just run and tumble. When running, flagella rotate in a counterclockwise direction, allowing the bacterial cell to move forward. In a Petricobacterium, the flagella are all bundled together in a very streamlined way. Interesting. They're just. I guess that would make sense that they all get bundled when there's this many flagelli. When tumbling flagella are splayed out while rotating in a clockwise direction, creating a loop motion, preventing meaningful forward movement, but reorientating the cell towards the direction of the attractant. When an attractant exists, run and tumbles still occur, however, the length of runs is longer while the length of the tumbles is reduced, allowing overall movement towards the higher concentration of the attractant. When no chemical gradient exists, the length of runs and tumble are more equal and overall movement is random. Yeah, I guess Sudoku is like it probably. Sometimes I wouldn't be able to do like a crossword puzzle in a car. I feel like it takes too much of my brain power to think of all the trivia, and it also just takes way too long. A crossword. I usually like if I do a crossword, it's taking like the whole day because I'm pretty slow. Look at it. They splay out. I kind of like it. It kind of looks like that gif of like Squidward when he's dancing. He just like it's all moved. It's all moving everywhere. And then it's all bundled when it's moved counterclockwise rotation and then clockwise. It's so fascinating. What do you mean they're moving? It's kind of unreal to think about. Run, and then they tumble, and then they run, and then they keep doing this until they get to the thing that they want. And it's just kind of cool because of the fact they're moving just based on a gradient. It's not like they're moving. They're not moving like how we move, like we walk and we're just walking and walking. They're moving because there's stuff along the way that they want to get to, and that will eventually get them to the where they need to go. Which maybe is kind of like how we move. We're motivated in the end. There's a thing attracting us to move. Okay. Yeah, and it's not like they're thinking either. It's again what I was saying about like you almost want to attribute this to being smart. Like they are like these bacteria are cognizant and they are trying to accomplish something, but it's not really. There's something telling it to do this. I always think that discussion is pretty fascinating. You hear that discussion a lot with cancer too. People want to believe cancer is this really smart creature. Like it's like they humanize cancer. Say it's smart. It's like blocking all of our therapeutics. It's working around all our therapeutics. It's trying to like kill us all, and it's so advanced. And I can understand how people get to that point of trying to think like that because sometimes the things cancers do to avoid therapeutics, it's like, whoa, how did it like evolve to like do this mechanism? But it's not. It's just you give it pressure and it's going to evolve to avoid that pressure. Because the only thing it's quote unquote thinking about is surviving. That's what all these things are doing. They know they have to survive and they're going to survive and evolve to survive. They're going to evolve to survive. It's nothing like really intelligent about it, but also it makes you like it's hard not to think of it as intelligent still. It's just it's so sophisticated. and it's hard not to like admire it. Okay, I think, how long is the rest of this chapter? Okay, this chapter is kind of long. Okay, well, we did get through the prokaryotes, so it's not like we didn't do a lot. We got through 30 about 50 pages. 50 pages today. That's pretty impressive, you guys. We got through 50. I yapped through 50, and I mean that only took 4 hours. Surely getting through there's 1,221 pages. We'll definitely get through this eventually one of these days. There's still so much interesting science we haven't gotten through. We're still on the learning the basic learning blocks. We there's like chapters in here that I think are really fascinating. I haven't actually read them. I haven't read them, but there was cool chapters about like the clinical applications of microbiology, which I'm excited we'll get to. You know, maybe it will take 5 years, but I think it's kind of neat. It's I'm learning and you get to learn, maybe, kinda, sorta. Or you can listen to just me. It it's like a nice refresher to go through basic biology. Sometimes I realize I don't know the whys as much as I would like to know. And not knowing the whys can be pretty bad. I think when sometimes you're arguing with somebody online, not that I argue online with many people, but if you get into a discussion with somebody who might be maybe perhaps a little anti-science, maybe just a bit of anti-science information, when you don't know the whys, you can end up getting into like you can get into some like bad corners because they just don't you need to educate them on like the basic fundamentals of science. It's like we were talking about in the beginning of the stream. The people that made videos about how they would like cover up their mirror with like a blanket and then record a video saying like, how does the mirror know that what I'm doing behind this blanket? And it's like they don't understand that a mirror is not a person or a thing, like a living thing. It's just a reflection of light. And so they're just completely unaware of basic science, like fundamentals, physics fundamentals, and they think the mirror like has the ability to know or see or whatever you want to say. Yeah, like I think that's when things like knowing, being very comfortable and knowing the most basics of science can come in handy. So you can explain gently, like, "Oh, sweetie, that's light reflection." I had a discussion earlier today about the extinction of most of Australia's megafauna 10,000 years ago, or whether or not humans or climate change were the primate cause. It wasn't interesting because they seem to have a somewhat rigid mindset. And yeah, that is one of the bigger problems, too, is sometimes you can engage in these discussions with people who maybe aren't going to change. They're not here to change their mind. And no matter what you say, you might not change their mind. They might be firm. They might be firm in their stance no matter what you say. And that happens a lot online, unfortunately. It I I don't like engaging a lot in like online discussions because of that. I think a lot of times people they're not here to learn, unfortunately. And it's usually easy to recognize that, though. It's pretty like obvious when somebody starts they're discussing not to like have a discussion, but they're just here to argue their stance and get their stance like seen. Discussions, like I think especially discussions on Twitter, they're pretty like they're not very conducive to like scientific communication because of that. There's a lot of bad faith actors on social media. The greatest extinction machines ever. Oh, that's 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 a pretty that's like such an extreme term. I don't know if I would want to say that. I think I understand maybe where that person's coming from, but it's not a very positive mindset. Let let's get out of the no, never mind. I was gonna go to the chatting section, but I don't think it doesn't matter. Yeah, it's more like I get where they're coming from, but it's like such a negative mindset. And then also the problem usually is when someone is like that negative or biased already, then it makes having discussions beyond that point maybe it's a bit more difficult because they're kind of clouded in a way. It's it can be it's so hard to discuss science. And it's not just like barring like discussing science with people who are different levels of science, like science expertise. It's just hard like discussing something, any kind of science usually. I guess that's why scientific communication is kind of like in shambles. Nobody, I feel like nobody, it's really hard to get it right. And I think especially in modern day, like with our like social media, internet platforms and such, no one's doing a great job. It's hard to find like an acceptable way to talk about science to a vast majority of people. Mm-hmm. Science is rough. I like talking about science though, and I always try to do my best to explain it as like new neutral to positive as I can. Maybe leaning more towards positive. I'd rather be more positive than negative about science when I talk about it. Depending on the subject. Mm-mm. Okay, I think I'm gonna wrap it up here because I realize it's gonna be 12 a.m. now and I have to get ready for bed. Yeah. Anyway, thank you guys for joining. I will be streaming tomorrow. It'll be an art stream. So I will just be drawing and yapping about whatever. So I hope you can join. And so thanks again for hanging out. Yeah, you should. It's bedtime. It's bedtime. Time to wrap it up. Okay, I'll see you guys. Bye bye.